I hope all the judges can hear me clearly.
Yes.
Okay, thank you, sir. So I'll just check there. So Anvish, please inform us when you go live. We're live. Okay. Welcome back, everyone, after the lunch session. We are now going to have the post-lunch session of BioBlitz 2021. We are going to continue with the presentations as we're going ahead with. So now we have the fourth presentation of the day. And the fourth presentation of the day will be given by Ananya Natarajan, Suhas Sehgal, and their teammate Mahima Samrek from ICER Mohali. So Ananya, Mahima, and Suhas, it's all yours now. The platform is all yours. Could you please share your screen?
Yeah, I'm sharing it.
Yes, it's visible. It's in full screen mode. Great. So we'll start with the presentation now. You'll get, as usual, 20 plus 10. So 20 minutes for the presentation. If you're presenting the bonus question, 2 minutes or 3 minutes extra. So we'll start. So your time starts now.
Hello everyone, I'm Suhas from ICER Mohali. So for the first question, we were supposed to test the hypothesis that in this hypothetical arachnid, the post-copulatory cannibalization happens and females eat the males. Next slide, please. Next. Now to test this, we have a null hypothesis and an alternate hypothesis. Our null hypothesis is that the males are not cannibalized post-mating. And our alternative hypothesis is that the males are cannibalized by the females. Now, to understand why this happens, the proximate reason of what we understand is that this probably happens because the females need nutrition right after mating. And to test this, we have another null hypothesis, which is that the male cannibalization does not affect the fitness of males. So the females need nutrition to increase their fitness. And the males also increase their fitness by dying. And it happens irrespective of the nutrient availability and their number. And the alternate hypothesis is that the male cannibalization affects their fitness, and cannibalization by the female depends on the nutrient availability and the number of males in the environment. Next slide. So a typical life cycle of the spider is like this. The females lay eggs, and the eggs hatch into spiderlings. They are immature, and then they undergo several molts, and they become sexually mature adults. So in this spiderling stage, they don't reproduce, and in the adult stage, they reproduce. So we would observe when cannibalization happens the most in, for example, in which stage the cannibalization rates are the highest. Next slide, please. So now we come to the experimental design. So we would collect 9 natural colonies of juvenile arachnids, and they should be collected from identical environments and should have identical sizes and sex ratio. Please note that these are, right now, these are juvenile. Next, please. These 9 colonies will be divided into 3 colony sets. In the colony set A, all of the colonies will be provided with normal amounts of food that they get, the normal amount of prey that they usually eat in their environment. And at this stage, no further manipulation would be done. In colony set B, all of the 3 colonies will be resource deprived, and they would not receive the optimum amount of nutrition which they get in natural habitat. Next slide, please. And in colony set C, they will be in resource surplus. We would provide them with more prey than they usually need in natural habitats. Also, please note that these are all juveniles, and we would record the cannibalism rate, if there is any, in these juvenile colonies. Next slide, please. So we would assess the cannibalism rate and the sex ratio every day till the last molting stage. And after the last molting, we will do some manipulations. Yeah, next slide. So in colony set A, so in all of the colonies, we would first remove all of the males right after the last molting. So when they become sexually mature, all of the males will be removed from all of the colonies. And they will be reintroduced to their respective colonies after some time. So in colony 1 of colony set A, virgin males will be introduced and will be allowed to mate with virgin females. But as soon as mating gets over, the males will be removed so that there is no scope of cannibalism. In colony 2, the same thing will happen, but the males will not be removed. And we will record the cannibalism rate. In colony 3, the same thing will be performed and the males will not be removed, but we will introduce more males. These males will be collected independently. They will not be the males which were taken from the colony 1. These will be males from other natural sources, and they will be like extra males will be provided to colony 3, and we will then record the rates of cannibalism in all of these colonies. And the same thing will be done in other 2 colony sets also. Next slide, please. Next. So after this has happened, we will record the cannibalism rate, and we will also measure the parameters that are directly related to reproductive success of the parents. These parameters will be clutch size, offspring weight, offspring survival rate, and the reproductive success of the offspring in the next breeding season. Next slide. So what we expect? So we expect that the cannibalism happens because the females need resources right after mating, and that's why they eat the males. So females with lower resources should cannibalize more. So their cannibalism rates should be higher, and the females which are provided with more nutrients, which are in nutrient surplus, should cannibalize less often, and their reproductive success should be highest. Next slide. So these are the results which we would expect, that that among all of the colony sets C should have the highest fitness output. Their fitness assays should be the maximum. And among the 3 colony sets, the rate of male cannibalism should follow the order of BAC. B should be in B there should be maximum cannibalism because they were resource deprived at all times. A was in the proper resource amount, and C had a lot of resources. That's why we have this order. And in each colony set, the order of cannibalism should be that the third population of each colony set should have the maximum number of cannibalism, because in these colonies they had access to more males, and the least cannibalism should happen in the first colony of each colony set, because males were always removed from these colonies. And the reproductive success should also follow the same order. Next slide. So in conclusion, this experiment provides a direct link between the rate of cannibalism and its dependency on nutrient supply and mate availability. And the proximate reason for this is that the females require resources right after they mate, and this helps males to increase their fitness. Next slide. Now, for the second part of this question, to understand why gene inactivation might have happened in the prey species, we first need to understand what adaptive radiation is. So adaptive radiation is a process in which organisms diversify rapidly from an ancestral species to new species when a lot of niches open for exploitation. Next slide, please. So we can imagine that a similar thing might also have happened in the ancestor of this of the prey species of an arachnid. Let's call it species A. So so when the ancestor probably moved to a new environment, and when it discovered that a lot of niches in the new environment, then probably there were some niches which did not have a lot of microbial parasites in them, and probably there was no need to express those genes, and those genes were not selected, and they probably acquired neutral mutations in them. So neutral selection is probably responsible for the pseudogenization in this case. Next slide, please. Now to verify this experimentally, we are going to consider two things. First is that if this has happened once, then this can be repeated experimentally again. And the other thing which we are going to do is we would do a comparative study of the species which are closely related to species A. So what we can do is we can maintain a population of species A in sterile conditions in a lab for many generations. So because the conditions are sterile, therefore they will not require any antimicrobial peptides. So we can expect that those genes will not be selected and they will acquire random mutations in them. And after many generations, if we sequence their antimicrobial peptide genes, then we might find random mutations in those genes. Also, if you look at the genes that are homologous to those antimicrobial peptide genes that have been lost in species A, in the close phylogenetic relatives of A, and then if we find a positive correlation between the pathogen pressure and the number of active genes for a good number of these species. So pathogen pressure is the number of pathogens which are present in their environment. And if this correlation is positive, then this would give a strong support to this hypothesis. Next slide, please. The type of selection here should be neutral selection. And there is another possibility. Next slide, please. There is another possibility. We have actually not given a full hypothesis to experimentally test this, but the other possibility is that the pseudogenization happened probably because the physiological costs of producing the AMP products were very high and their benefits were not that good. So probably they evolved some other mechanisms to deal with the infections. Now to test this, we would just simply infect species A, a population of species A, with those microbes which are fought by cecropians. Now cecropians were silenced in the not silenced; they were pseudogenized in these species, and if they fight this infection and if the mortality rate is is insignificant, then we can conclude that they have probably devised new mechanisms of fighting such infections. Yeah, over to you, Anand.
Yeah, one second. Yeah. So the next question we are going to be dealing with is gut microbiota acquisition. So what was basically done was meconium samples were collected from neonates shortly after, within 24 hours of birth, I guess. And the presence of these particular 4 bacterial species was tested using fish. And BIF 164 was detected in 1 sample, BAC 303 in 3. ENC 221 in 5 of the samples, and lab 158 was detected in 7 samples using fish. And the cell count was estimated using QRT-PCR and using fluorescence, and the data was given for making a calibration curve, which was made by us using a linear regression least square fit for each of the bacterial species, and we calculated the number of bacterial cells per unit mass of meconium samples for each case, and these were the results. So there were 16 meconium samples, and as given, these were the bacterial cell counts for. And we can see the Lactobacterium seems to have the highest cell count, both in terms of number of samples in which it was detected and general count in each sample, followed by ANT and followed by BIF. BP data was not, we weren't able to get the data because there was, BP was not cultured, and so a calibration curve couldn't be made for it. And when we look at temporal variation of bacterial count with the meconium interval, meconium interval in this case was defined as the time between when the amniotic sac broke. So when the, in common parlance, the water broke, to when the meconium sample was processed till the very end of the processing. So that is the time interval which is defined to be meconium interval in this case. So this is the raw graph for total bacterial count versus the meconium interval. So as we can see, it looks like it seems to be pretty low till 2000 minutes and gives a sudden jump. But if you look at the log bacterial count, we can see it follows a more gradual pattern. So which is fairly reminiscent to the bacterial growth curve in this case. And you can see the lag phase probably corresponds to when the baby has just come out of it was given birth to, and as time progresses, it exponentially grows the bacterial count. So the model proposed from all these observations is as follows. So since the gut microbiome of the neonate seems to follow a growth curve similar to standard bacterial growth curve, and the number of bacterial cells were very low to start with, if you look at the BIF lab counts, I guess it's safe to assume that the microbiome of the neonate is not fully developed during gestation itself. So it's even, let's say, even if it had acquired very small quantities of bacterium during when it's growing in the womb, it has not the microbiome itself has not fully developed, because if it had been developed, there wouldn't be this exponential growth in the meconium sample. I'm assuming it will be more of a steady state with some fluctuations maybe. So a major portion of the gut microbiota was probably acquired during birth, when the infant would actually be exposed to vaginal microbiota. And there is evidence that vaginal microbiota actually has a very large diversity, and it's dominated by lactobacillus. So this is in fact a lot of papers will show you that this is probably one of the first times in which the infant is exposed to a environment which is not highly sterile, whereas the amniotic sac is a highly sterile environment, but once it breaks, it's exposed to an outside environment which is not as sterile anymore. So the main model we are proposing how gut microbiota has been acquired is during childbirth, when it's exposed to the birth canal, and it acquires it through the from the vaginal microbiota. So yeah. And other observations are that BIF 164 was only observed in one sample out of the 16. So it might not be acquired in large numbers during gestation or childbirth, and it's probably acquired either from the outside environment or through breastfeeding. And the other two were detected in 5 and 7 samples out of 16. So these are probably acquired at childbirth. And LAC has the largest bacterial cell count. So it is probably acquired in large numbers during childbirth, whereas the other two are also supplemented from breast milk and the environment. So what the main model we are proposing is that a large part of the microbiome is acquired during birth from the birth canal, and some components of the microbiome are also acquired during breastfeeding and from the environment. An alternate model which could hazily explain the data is that it might have acquired its microbiome from the mother during gestation itself, when it's growing from the placenta or otherwise, and after its gut has developed. But maybe the microbiome was suppressed from growing because there were some factors from the mother which suppressed the microbiome from growing. And after birth, of course, since there are no maternal factors supplied to the baby through the placenta, maybe through breast milk, yes, but not through the placenta anymore. So maybe the growth of the microbiome is not suppressed anymore. And we would prefer the first model over the alternate model, which is it acquires it through from the vaginal canal rather than during from the placenta during gestation itself, because the first model seems more parsimonious than the second one. The second one would postulate that there are maternal factors which suppress the microbiome growth, and there is no evidence for these kind of antibacterial factors produced by the mother. So the first model seems to be far more convincing. And with regards to gut microbiota and fetal neurodevelopment, so as we know, the mice were reared, SPF, GF, and ABX kind of mice were reared. And for observing the effect of microbiota on fetal neurodevelopment, two kind of analysis took place. One was measuring axonal volume fraction and corresponding paw withdrawal thresholds, and this is the data. So SPF, ABX, and GF. So as you go down this, you can see the microbiota population will steadily decrease, whereas the axonal volume also seems to decrease with decrease in microbiota population, whereas the paw withdrawal threshold is increasing, which shows that there is an decreased sensitivity as microbiota diversity decreases. So, and the other analysis was that thalamic neurons were grown alone or from endogenic cues from the hypothalamic explant, and an FACS analysis was done. And this is kind of like the Y versus X plot. I have just condensed the observations into this plot. So when the thalamic neurons were obtained from SPF and the hypothalamic, there was no hypothalamic this thing. There was some amount of X. But when
so just to interrupt, sorry to interrupt. So your time is already up. So since you are continuing to present, you may be facing a little penalty, but I think you can continue. Plus, just try to wind up in another couple of minutes maximum.
Sure, yeah. So based on these observations, this is the model I am proposing for how microbiota influence the neurodevelopment. Three factors, T, H1, and H2, is produced by the microbiota. T is a factor that acts on thalamic neurons and promotes the expression of X, whereas H1 and H2 act on hypothalamic explants. And H1 represses the repressor of X, and H2 activates an activator of X. So this is the model I propose for how the microbiota affects the fetal neural development. And this model would explain why on adding certain hypothalamic explants, the amount of X shifts either to a lesser or a higher degree. So with regards to Y, Y seems to be expressed at a constant and high rate in all thalamic neurons regardless of the microbiota status. So expression of Y is independent of microbiota. Yeah. Thank you.
Thanks, Ananya. So, and Suhas and Mahima. So, Ariman, did you track the extra time?
They took almost 2 minutes extra.
Okay, so you took 2 minutes extra. So now the judges can ask any questions to the team. Beautiful presentation. Yeah. So the judges can ask any questions that they want.
Yeah. Can I start?
Yes, sir, sure.
So in the spider story, you made the assumption that the female spider will eat the male spider because she's hungry or she's tired or whatever. So this is called a flexible character. But many characters are fixed. So it's a species-specific instinctive trait that the female will cannibalize the male. And if your species has this kind of a fixed character, how will things change? You can't design a new experiment now, but just in 1 or 2 minutes, can you tell me what that will mean in a species where cannibalism is fixed and not left to the choice of the female?
So if cannibalism is fixed in these species, then probably they will, like when we will remove the males right after the mating, then probably we should expect to see some behavioral changes in females. The female should either look for the male around in the environment, or
the observed reduction in the number of males, is it due to cannibalism, is the question you're trying to answer. So cannibalism is there, it is fixed, but the observed reduction in the males need not be due to that. It could be due to something else, or it could be due to additional factors, right? So you want to know that the observed reduction in the males is due to cannibalism or is due to something else.
Right. So basically, we are conducting this experiment in a laboratory setup. So all of the other reasons, like for example, if it is due to infection or something, then those things get cancelled because.
Yes, but there could be intrinsic reasons, right?
Yeah, so right.
Think, for example, that males may have a shorter lifespan, right? So there could be reasons intrinsic to the male spiders.
So
both in the
like in one of the manipulation which we did was that the males are removed right after mating. So if we observe that those isolated males die just by themselves without, like, they are not being attacked.
No, they will not die. If you remove them, they will survive. But if you don't remove them, the female will eat them.
Yeah, right. No, I'm actually not trying to understand. Like you said that they are dying after mating because of some other factor.
They are not dying, right? The female eats the male. So given a chance, the female will eat the male after copulation. And if you, of course, remove the male, there's nothing that the female can do. But given a chance, she will eat the male after copulation, with whether or not she's hungry.
Right. Now to test, now what I think is that if this is the case, if female would anyways eat the male, then she would probably search for male in the environment. And if we remove the male from the environment, then there should be some behavioral changes. Like the female should probably look for the male around.
Yeah, but what is the question you're trying to ask? I thought the question you're trying to answer is whether the observed reduction in males is due to cannibalism or not, or due to some other reason, right? That was the original question.
Right. So, like, if we detect a change in females' behavior, then that probably points to the possibility that the female, like, wanted to eat the male, and like that is why this happened.
Okay. Anybody else?
Yeah, thank you. Nice presentation, everybody. I will just ask you, it's very interesting concept you made. In colony 3, you have introduced extra male, right?
Right.
So now my question is, sorry?
Yeah, continue.
Yeah. So the question is that what was the motivation or purpose for you to add extra males there? Were you trying to understand that whether the number of a female, how many males can the female feed on, or you are wanting to understand that whether if the male are introduced from some other territory, whether the female is able to sense and kill them?
No, actually, the actual motivation was that is there any bias in feeding patterns? So after mating, does the female prefer males over their prey? Actually, we didn't include this here, but this is what we wanted to see. For example, if females eat males even after eating their mate, then this means that they have some bias, some food bias, right?
So,
And probably the males
okay.
might have some factors in them which are beneficial for the females. And if males allow females to eat them, then and if those factors get into the female, then probably that is benefiting the males because that would increase females' reproductive output. Assuming that those factors are good for females.
Okay. Okay. But the reason that you're adding extra male, was it to understand that will the female eat the male that it has mated, or it can eat any male from any other colony? That was the motivation? No, was that the motivation for the experiment?
Yeah, that was the motivation.
Okay, fine. My next question is for this bacterial, comparing the bacterial growth curve with the consortium which is present in the sample. Yeah, this one. So now, if you really understand this Meconium interval versus the comparison with the bacterial growth curve, normally this kind of growth curve you obtain with a monospecies or say a pure culture of bacteria. How can you comment on the consortium which it is not about the number, but it is about the diversity of the species, right? So can you comment on, or can you compare this growth curve of a single species, which actually when it happens in the tube, versus the gut flora, which is very diverse?
So I guess I kind of compared them because I was operating on the logic that let's say you had gotten like very few numbers of bacteria at birth from the vaginal canal. So each of the species might individually follow a bacterial growth curve similar to this. So assuming there were very few number of bacteria introduced in the first place. So I guess I did kind of assume the aggregate will also follow a similar pattern, because I'm assuming that all of them will follow their own individual bacterial growth curves, and the aggregate might look something similar.
Have you not taken into account that they also can outcompete each other, or they may have some kind of a bacteriostatic effect on each other?
I have not accounted for that. Yeah.
Okay. Thank you.
Thank you.
Yeah.
Very small question. Pseudogenization, can you people predict it just by comparing the sequences for say near by species? I mean species, Q species, whether pseudogenizations happen or not? Experimentally, how you will prove it?
So can you repeat the question? I didn't understand.
I mean, if you compare the say few species, the sequences, right, which are very close. I mean, species producing the sab eating, right? So the microbial checrophin and diphtherin, right? They were pseudogenized, right, in this condition, right?
Right.
So if you compare now with the all the sap feeding organisms,
with their relatives, with the related species.
Yeah, related species. My point is that, yeah. So if will you be able to deduce something out of it experimentally?
So actually, what we thought was that if this gene silencing has not happened in those relatives which have a lot of parasites in their environment, and in some other relatives, if this gene silencing has happened and in their environment the parasites are not much abundant, and like basically if you get a positive correlation for a positive correlation for gene silencing with less number of parasites in the environment, then this would give a good support to our hypothesis. This was the idea behind this. So basically, the gene silencing, the neutral evolution thing has happened with other close relatives also. This was the idea basically, depending upon the environment, because selection would happen. Selection would change with the environment.
Okay, right. For the alternate model in the gut microbiota thing, where during the gestation the infection is happening, right? Yeah. So how you will prove that the alternate model is right or wrong?
So one thing
Any
I can
kind of-
think of. One thing I can think of is firstly, I guess there is really no support for this, even from when I try to find literature for this, in terms of like if this would even happen in the first place. But I think a first stage would be whether looking at the genome of the human or the mother and seeing if there are any antimicrobial peptides which would work against the gut microbiome, if there are presence of such proteins. So if those are present, then we can think about how to test for whether such factors exist that suppress the microbiome. So I guess the first step would be to see if there are certain maternal factors which are antimicrobial peptides and work against the bacteria and the gut microbiome.
Not necessarily. You can just look at some of the abortus also in the during early gestational first trimester or second trimester. If there are individuals who have been aborted child, right? You can see if you can take some of their gut microbiota if they have got it or not, right? That is one way.
Right.
Another is that you can use the animal model also, right? And this is just for your knowledge. I mean, the only 1 or 2 months back, the Cell paper came out. First, the child gets the first microbe by second trimester. Okay, the start of the second trimester, and it was a Cell paper.
Right.
Okay. Right. Okay then. Yeah. Good
Thank you.
presentation. Thank you.
Thank you, judges, for the presentation. It was so Ananya and others. It was a lovely presentation. Just one quick question I have on the antimicrobial peptides. You mentioned about the specificity of secropins. Now, secropins are very broad spectrum, and they are active against all gram-negative, gram-positive, and also even some fungal pathogens. In that case, how will you check?
So, like, what are you asking? What would we check? Like,
Just the next slide. Another possibility. The next slide. Yes. So here you have mentioned about the genes that have been lost by a secropin. We can try to infect some individuals with pathogens that are usually fought against by secropins. Right. Now, those are all pathogens. So will that not implicate a fitness cost? Because all pathogens, a huge spectrum of pathogens are fought by secropins. If you talk about some diptericins, like those which are diptericin, which fights the Providencia redgeri kind of thing, those are highly specific. But secropins are very broad spectrum. So in that case, Yes, go ahead.
Yeah. So what I want to say by this is that they might have evolved some other mechanism which is not very costly in terms of physiology. So what we would do is we would like you're saying that cecropins fight against a lot of different pathogens, right? So I'm not saying that they have lost that ability completely. They have evolved new mechanisms which are not very expensive, and but they have the ability to fight these infections. That's what I wanted to convey.
Okay, thank you, Suhas. So in case, just if you could come up with some concrete example of what could be such a mechanism, you could drop it in the chat box. We'll not waste any more time.
Actually, this was a side example sort of thing. The main hypothesis was the
Yeah.
Okay,
thanks.
so thank you. So now we'll go over to the next presentation. The next presentation will be by Anurag Chitawar. Anurag is from Yogiraj Public School. So Anurag, are you here with us? I think. And can you hear us?
Hello. Just
Yeah,
a second.
so-
Yeah.
Yeah. cool.
Hello.
Yeah, you're audible.
I'll start sharing. Just a second. Can you see my screen?
It's still loading. Just in a few minutes, probably we'll be able to see.
Okay. Can
Yeah,
you
it's
see my
visible.
screen?
It's visible now, Anurag. So you can
Okay.
go ahead. So your time starts now.
Okay, awesome. So let's just start with the first question, ecologist's wonderland. So the null hypothesis that I propose for the experiment is that female spiders do not engage in sexual cannibalism. And the alternative hypothesis, of course, being that the female spiders do engage in sexual cannibalism. Of course, there could be other reasons for the male's disappearance, such as the fact that there could be certain male-specific diseases that specifically attack males, or there could simply be like a recessive disease that would, again, disproportionately affect males. There could also be predation. These would not be looked over in like, we could not give an answer in the positive from the data from my experiments alone, but we will get an answer in the negative of whether sexual cannibalism occurs or not. Okay, so some characteristics of the spider that hint at a sexual cannibalism. So the fact of the matter, that the species is heavily dimorphic, the female is much larger than the male, as is the case with many other species showing similar patterns in spiders in particular, where the female is much larger than the male. Also, the males are monogynous. This could be explained from the fact that they're likely to mate only once with the female, because well, they die after their encounter, making them effectively monogynous. Right. So why do the spiders eat the males? Assuming that that happens, a null hypothesis could be that the cannibalism of the males does not lead to a greater reproductive success. The alternative hypothesis, of course, being that the cannibalism of males does lead to a greater reproductive success. So some possible explanations for the alternative hypothesis. So the males that get cannibalized get to copulate longer, ensuring a deposition of a greater load of semen, as copulation times are often generally associated with greater paternity, certainty of paternity. Secondly, cannibalism could also ensure paternity by providing the female with nutrition so that the clutch sizes are larger and the brood survival rate is improved. Right. So for the null hypothesis, this is of course only for the case of post-copulatory cannibalism. In the case of pre-copulatory cannibalism, there could simply be the fact that males are suboptimal, or they are mistaken for prey, or there's like a simple aggression spillover where there is no discrimination between the fitness of the male, or there is no mistaking the male for a prey. It's simply a spillover from the aggressive behavior of the female in the case of pre-copulatory cannibalism. Yeah. So let's go to the experiment. So for the experimental protocol, each immature female web location was marked and then receives an individual number. Additionally, the distance to the nearest female neighbor is measured, and every day the number of males staying close to the subadult female's webs or in their web is noted. So during daily controls, the developmental status of each female is closely monitored. The removal is done in a way so that there is no disturbance to the males that are already present near the web. So what this ensures for us is that we collect the subadult females shortly before their final molt. In the laboratory, the collected females will be kept in individual frames where they build their webs. All spiders will be fed the same food, which can be basically gleaned off of through a cursory study. This could also be a potential site of error in the sense that the food we provide could possibly lead to a modification in the behavior of the spider. So when the females molt to maturity, they are individually marked with a unique combination of color dots. Then the females are returned with the frame to the original website, and the frames were attached to the ground with tent pegs. Yeah. Sorry, my presentation is so bad. Yeah, okay. So the females are then observed. Right, so once a female, if a virgin female does not copulate on a given day, she's carried back in her frame to the laboratory so that all copulations are ensured to be in the be ones that are observed by us. After having mated at least once, females are left in the field so that a natural occurrence of virgin and mated females occurs, and there is no artificial change in the mating strategies of the males due to the artificial abundance of virgin females over mated females in that case. So when a male visits a female, we note the following about him: his size and number of legs, the time of his first appearance, the time of his first contact with the frame, contact with the web, contact with the female, beginning and duration of courtship and copulation, and the occurrence of sexual cannibalism or male escape from a female attack. What this allows us to do is make a sort of inference about the male's fitness, whether the female recognized the male as being a mate or recognized him as a potential prey, or if there was a simple aggressive spillover based upon the attack the duration of female attack to the duration of his first contacts. Okay, after having mated at least once, the females are left in the field. So as to maintain, again, okay, that is a repetition of the primary sentence. That is error by me. I apologize for that. When males survived the copulation and left the web, they were captured and marked with a specific color dot before they were released again. In this way, we could identify the males that have been attacked once and have left. A subset of the killed and wrapped males were removed from the web for further analysis as well, such as the amount of semen deposited by them into the female. Some sterile males were injected into the population to measure the effects of paternity on the paternity percentage that cannibalism could have. What we are doing here is like a more loose and less rigorous sort of a version of like a sterile male method. So sterile males are injected, and then through the process of subsequent copulations between normal and sterile males and the patterns of them being eaten or escaping, we can effectively measure the effect on paternity that the cannibalization has, as the copulation done by the sterile male would show up in the form of undeveloped eggs. For this, we have to establish controls such as cases where the female only mates with normal eggs, so that we can get a baseline normal reading of the undeveloped eggs in a normal sort of mating. Anything above that we can then directly attribute to the sterile male. Yeah, so that is that. So moving forward, so some expected results. So by looking at this population of unmarked males and marked males along with the observing of the killed males, we can determine whether cannibalism is a usual occurrence of significance or not, and whether it plays any sort of a role in this depression seen in the population of males. We could also potentially calculate the proportion of males that left without mating, and whether this was due to female disinterest and or mate choice, to make inferences about the presence of mate choice within these spiders. This could be done because of the extra data we collected about the male's fitness and the times of contact and copulation. Multiple copulations can be easily mapped out along with the relative rates of males getting eaten. So when the first or the second or third male, is he more likely to get eaten, is he likely to get eaten, and so on and so forth, and the copulation time, so that the first, second, third male, how does the copulation time change with repeated copulations? And with inference, and again, we can make inferences about whether these behaviors are linked with maximizing paternity. We could also look at whether female mating success was the product of spatial and temporal factors during the mating season. So it is a seasonal sort of mating. The mating season, they don't mate throughout the year, they only mate in the monsoons. So we could look at things like humidity and sunlight and spatial factors like territory quality and whatnot to again make inferences about whether how the female's mating success changes with these factors. Another thing that could be done is by artificially inducing a gap between the male's killing and his getting eaten. We can make inferences about whether the behavior is primarily nutritive or is it primarily about the killing act of killing itself, that the female seeks out this behavior in the case of postcopulatory uh, cannibalism. Yeah, so that's that. Moving forward. So some possible sources of error. So by removing subadult females from males for a few days for the web making, there is some interference with male mating strategies, which could have caused an increase in male mortality, influence the sex ratio, causing an underestimate of female mating rates. Mated females remained in the field so as to keep a natural virgin to mated ratio, but during that time they were left unsupervised. Though this can be minimized by optimizing the time the data is collected with the time of the greatest activity of the spider, so that such an error is minimized. Yeah. Okay, so that's that. Some proximate causes of cannibalism. This is for postcopulatory. The male makes for a nutritive meal, provides nutrition needed by the female for the child rearing, thus leading to larger clutch sizes, larger offspring survival. Cannibalization of males could ensure longer copulation time for the male. So ensuring paternity for the male as a longer copulation time generally means greater semen transfer, which in turn means a greater chance at paternity. These spiders are generally internally fertilized. So yeah, that is there could be other hypotheses which we haven't considered. Let's move forward to the aphids. Why would the aphids pseudogenize antimicrobial peptides? So my hypothesis is that the pseudogenization occurs primarily to be a better host to these symbiotic microbes that are found in the aphids that help them with gaining these essential amino acids, along with helping them with coping with the high osmotic rates, high osmotic pressures of the phloem and the high sugar content, as is mentioned in the slide. The experimental setup. So what you could be using, since the genomic sequence is available to us, is a Ka by Ks test. A Ka by Ks test would compare non-synonymous mutations to the number of synonymous mutations, and the ratio would indicate whether it's a directional, neutral, or negative selection. A ratio of less than 1 would indicate a negative selection, not a stabilizing selection. That is an error on my part. A possibility of stabilizing selection cannot be rigorously ruled out with this setup, but it is highly unlikely considering pseudogenization is a loss of mutation event, and a loss of mutation event is unlikely to be heterozygotic. So the comparison can be made between the gene sequences of aphid and its close relatives to sort of determine that pattern. Another setup could be done on a primarily phenotypic basis, wherein we artificially inject certain amounts of AMPs in varying doses to different groups to measure the effect it has on the fitness of the aphids. If there is a consistent decrease, we can say it is a directional selection. If there is none, there is a neutral selection. If we see 2 poles, it could indicate a disruptive selection. There's of course the problem of the AMPs getting metabolized when injected, which could be a major cause of error in such a study. Okay, for question number 2.1. Possible hypothesis could be that the infant is born with a gut that is sterile, followed by rapid colonization by microbiota, with the first inoculation being through the vaginal wall. The alternative hypothesis being that the infant is born with a gut not sterile. The gut microbiome could get its first inoculum through the swallowing of amniotic fluid and through the placenta. The other sort of colonizations remain the same. The data says that the former hypothesis is much more likely. It is better supported by the data to sort of temporarily map out the population of bacterial cell counts, and with the time we defined the Meconium interval. This is the total time from the externalization of the amniotic sac contents to the time of parturition, plus time to passage of meconium in the laboratory, as well as any further processing. The start of this interval would coincide with the commencement of colonization if a sterile amniotic sac is true. So what do we find? We find that there is a rather strong correlation between the MI and the bacterial counts that were observed. So the microbial colonization of meconium is strongly shown to increase with time, which is true for both of them. However, there is a very low bacterial cell counts identified for low meconial times, and composition of the pioneer infant microbe also supports this hypothesis. 8 of 16 tested for bacterial populations using fish showed no appreciable presence of fluorescence, and the rest showed low approximate cell counts. So here is the data for the Meconium interval with the cell counts. The cell count is logarithmic. So yeah, there's that. You can see a very clear sharp increase here with time. Beyond that, we don't really have data from the 0 to 250-minute mark. That would also provide a great load of information in terms of saying conclusively whether the gut is sterile or not at birth. Here is the approximate bacterial counts for the 16 pioneer microbiotas. The bacterial counts are approximate because we did not have a fluorescence response curve for the BP group, BP probe. So 8 of the 16 of these show no fluorescence whatsoever. Yeah. Okay, so that concludes question number 2.1. Moving on to question number 2.2. So does maternal gut microbiota influence fetal neurodevelopment? The answer is, well, yes, it does. So the proposed model for this is as follows. The thalamic neurons of embryos from ABX and GF dams show a decreased expression of X along with a decreased external volume. In ABX and GF rats, there was also a decrease in sensitivity to the paw withdrawal test, suggesting that the microbiota depletion result in impaired tactile sensory behavior. As we can see here, the paw withdrawal threshold increases with the mean for the paw withdrawal threshold increases from SPF to GF and ADX. Similarly, for the axonal volume, there is a sustained decrease from SPF and GF and ADX. Here, the mean. So the proposed model states that the presence and absence of microbiotas in these mice mothers affects the intrinsic factor of the thalamic neuron development, as is seen here. The SPF neuron and the ABX and GF neuron show significantly different expression patterns for the X gene in terms of, as is seen with the different fluorescence for X here. Comparing SPF GF and GF SPF, we see that the germ-free lineage has an impaired response to hypothalamic factors. This is also seen by the fact that ABX SPF shows an intermediate response to hypothalamic induction, while GF shows no response. So as we can see here, SPF gets induced by the hypothalamic endogenous signals of GF to produce a GF-like phenotype, while in the case of SPF in doing the induction, the GF does not respond. Comparing ABX SPF and GF SPF, we see a decreased response to endogenous factors in GF in comparison to ABX, with the ABX neuron showing an expression pattern intermediate to that of SPF SPF and GF SPF, as is very clearly seen here. In the ABX group, they were weaned off of the microbiota in the middle of gestation. The germ-free group had no microbiota to speak with from the beginning, while the SPF group was normal. So this could sort of explain how the data shows an intermediate sort of an induction as semi-intermediate induction, as is seen between ABX and GF in the none case, and the sort of intermediate case is as is seen here in the ABX SPF, GF SPF, and SPF SPF case. So yeah, my hypothesis is that the gut microbiota regulates metabolites that enter host circulation, which in turn play a role in influencing host and offspring physiology. So certain metabolites from this microbiota affect the receptivity of hypothalamic neurons to endogenous signals, and also play a role in reducing the role in regulating the expression levels of X in these neurons in the absence of endogenous signaling. So tissue-derived cues are necessary but not sufficient for mediating this microbiota-dependent changes in X expression, and suggest that the depletion of the maternal microbiome impairs this complex interaction that can regulate thalamic neuron development. There is also a decrease in axonal volume and sensory response. X might have a role to play there, but again, we cannot state that in a concrete and rigorous manner. This might just be something that is simultaneously affected by an effect of the microbiota on the neurophysiology. Again, so what can I infer about Y? So Y is not affected by the presence of hypothalamic endogenous signaling in this case. Again, this cannot be completely ruled out because this could very well be due to the particularities of our experiment more so than it is about like a normative statement about Y's effects on hypothalamic endogenous signaling and Y's effects. Y is also not affected by the presence or absence of gut microbiota with reference to its expression patterns, like its expression, sorry, I meant the expression numbers. So the fluorescence, Y fluorescence remains the same. There could be, however, a change in the spatial dynamics or in the function of Y, which is again not something that has been studied through the data given. Y is a protein exclusive to murine neurons, as was given. With that, I conclude my presentation. Yeah, that's all. Thank you so much for bearing with me.
Thanks, Anurag, for the presentation. So now we can have some questions from the judges.
Oh, okay. Can I go with the question?
Yes, ma'am.
Deepshika here. Yeah, very nice presentation, Anurag. Would have been, you had a lot of sentences. Would have been nice if you would have put it as a model, but even though your all experiments and your hypothesis very beautifully shown. If you mentioned that in these antibiotic A, B, X, and GF germ-free, how important is a gut flora? And then in these ABX and SP, GF mouse, the power withdrawal time increases. Now, if you really want to prove that in these mice you can revert the situation, what kind of experiment would you put?
For a reversion, what we could possibly do is an artificial sort of an infection or colonization of the gut microbiota post their post parturition, like immediately post parturition. We put them in a sterile cabin, sterile sort of an environment, and then inject the necessary microbiota in the first few stages of their development to then measure the paw withdrawal thresholds in comparison with others that had others that were simply read in the form that they were, so to speak, if that
so what
sense.
kind of microbiota would you like to introduce and how?
Okay, so I guess what we could use are the microbiota from the SPF, since they provide us with, like, again, a singular sort of a metric to measure everything by. So we take microbiota samples from SPF through fecal matter maybe, and do like a fecal matter transplant or something like that.
Okay, yeah, you can. Good. My next question to you is, see, in that in the experiments where now it is a big challenge to show that whether the cannibalism of the male is because of the food, or there is a some other purpose that the males are killed. Now, if you really want to dissect these two pathways, that is it really for the food and it is not for something else, what other experiment can you think of?
Okay, so if we maybe what we could do again is to ensure that it's not really about like the food itself, is create a certain, I mean, it's a species in the wild, so I'm not sure how much artificial sort of like lab culture we could possibly do with this, but if possible, like something like creating lineages that are artificially sort of like smaller in size and smaller in just in like terms of their biomass, right? Again, we get like the very clear data about mate choice from this experiment. So we could use that in conjunction with the sort of artificial males which we present to the females to sort of weigh the two competing there and check whether it's really for the food or really something else that is affecting the.
Okay, thank you.
Thanks, ma'am. The other judges, any questions?
If I can just continue with
Yes, sir.
your answer to this question. If we want to know whether cannibalism is for the purpose of satisfying food requirements or not, wouldn't it be easier to have the females well fed in one case and hungry in another case, and see whether the incidence of cannibalism will be different between these two cases? Do you think that will work?
If the females are hungry and if the females are well fed,
Yeah,
that should work. Yeah, that should work. But
might be easier than
again,
lineages, right?
Yeah, but again, we'll have to control for the male sizes as well simultaneously. But yeah, it would be a lot easier to do and a lot more achievable than what I propose.
The difference is that in one case the females are hungry, in the other case they are not hungry.
Mm-hmm.
Then everything else is the same. Then the difference between these would help answer that question, isn't it?
Yeah.
I'm trying to emphasize that it's good to begin with the simple experiments and then go to the more complex ones.
Also, I think here we could also get a better idea of whether if the pre-copulation or the post-copulation cannibalism dynamic, sort of like how that plays out as well, I feel, with the hungry and the well-fed ones.
Exactly. My other point for discussion was many of your plans are like things that you do in the natural environment rather than the laboratory situation. So the advantage is that this would be more natural, but have you thought about how much more difficult it would be to do this in the field and how much disturbance you might cause to the animals when you start making measurements and look at all of this? So there is a kind of trade-off between a lab experiment and a natural field experiment. It's not clear that one is always better than the other. I was wondering whether you have considered the difference between a lab experiment and a field experiment.
I see. So like my older model before this one was primarily completely lab-based, but then it sort of just struck me in terms of just like looking at the question again. So the species is a newly discovered species, right? So there would be a rather unique sort of challenge in terms of just like cultivating that species and ensuring a environment that's even sort of like comes close to approximating sort of environment that would make sense, naturally speaking, right? Of course, these experiments, they are very extensive, and again, the experiments themselves, the extent to which these could be done would depend upon how large these colonies are, what sort of a distribution the spider itself has, the places it lives in, whether they're accessible or not, and whatnot. So yes, definitely there is going to be a big challenge there, but I do think that a lab experiment would be much tougher considering the species is not well known to us.
Okay.
Can I ask one simple question? Right? Yeah?
Yes, sir, sure.
So Anurag, very good presentation. So can you think about how population study of, say, young kids, right, could help in understanding that gut microbiota are involved in the neuronal development?
So again, so like human kids or like rat kids? Hello? Sir, I think your video got paused.
Yeah, I think we lost connection.
I mean,
Hello?
in the...
Yeah, you can
Hello?
continue,
Yeah.
Anurag. Yeah, what you wanted
Okay.
to know, whether it is?
if you're talking of like humans or rats, because in the case of rats we can be a lot more invasive, right? And with humans we'd have to.
I think
he's-
Professor Anantamba is back now.
Yeah.
My internet is coming and going. Can you hear me now? Just,
Yeah, sir.
Yes,
I can
sir.
hear you perfectly now.
You can just do it, let's say in the human, people have done it in the human extensively.
Mhm.
You don't need to get into rat. How population study could deduce that the gut microbiota is involved in the neuronal development?
Mhm. Again, I guess fixing of a geographical location, so taking a very specific subset of the population through very strict inclusion-exclusion protocols, as was seen in the microbiota study before. Very strict inclusion-exclusion protocols, very limited geographical boundary that is generally like culturally, culturally and like physically similar across its domain. That would first of all help us sort of get rid of any sort of external variance there. Beyond that, we could, in that sort of environment, then we could look for edge cases, in particular with the gut microbiome development and say changes that occur with vaginal or cesarean, and then look at the changes in the communities that produces, and then look through certain say metrics that like the Powell-Rowell test, whatever like the human equivalent for sensory motor, tactile sensory behavior, it's R, something like that.
I understand what you are trying to say. Yeah, but yeah, what you talk about cesarean, non-cesarean, they don't so much, but the people have tried comparing the gut microbiota and the autistic children, right? People with the dyslexia and things, right? Okay,
Yeah,
it's
that
very
would
well known.
also, yeah,
Right.
that would be a very particularly
Yeah.
good.
Okay. Thank you. Right.
Thank you so much, sir. Thank you so much.
So thanks to all the judges for the question. We are about 9 minutes into the question. So if there is any question from the floor, a quick question, we can take it. YouTube chat, anywhere here, anyone wants to ask any one quick question? Anyone? Okay, seems like there are no questions. So thanks, Anurag, for the presentation. It was a lovely presentation. So now we'll move over to the next finalist team of the day. And the next team to present is Shraman, Adrija, and Tisha from ICER, Kolkata. Are you guys ready?
Yeah.
Okay, cool. So
I'll just start presenting and
yeah, sure.
see my screen.
Yeah, so Anurag, can you please unshare? Yeah, thank you.
I've unshared. Sorry about that.
Yes, thanks.
Yeah, is my screen visible?
Yes, your screen is visible. It's now full screen. Yeah, so if you guys are ready, we can start. So your time starts now.
Yes, hello everyone. We are the team from ICER Kolkata, comprising of Shaman Jana, Tisha Das, and myself, Adrija Adhikari. So for the first question, we have framed our null hypothesis as the female of the species, Arachnidus bioblitzia, does not cannibalize or eat the male with whom she has mated after mating. So the alternate hypothesis that we will be testing for is that the females of the spider do kill or cannibalize the males that they have mated with after the act of mating. How do we, so the first experiment that we were required to do is to test whether cannibalism actually takes place in this spider population. So what we have done is we've kind of had a setup. So setup 1 consists of 5 female spiders only. Setup 2 consists of 5 male spiders only. And setup 3 consists of 1 female spider and 4 male spiders. So all of these figures are made with biorender. So what we do is we tag the spiders to monitor the activity and to actually figure out if cannibalism occurs or not post-mating. So why have we taken 3 setups? Because there might be causes of natural death, there might be accidental death, there might be pre-cannibalism that we could tag, which is what the tags do, so that we can monitor if cannibalism only occurs after mating, or is there a significant probability that the spiders may be eaten up before the ritual of mating. And what we do after that is we monitor the tags, and we see that if the males of Arachnides bioblitzia are actually killed after mating. So if the number of males in setup 3 are significantly less than the number of males, and if we monitor the tags and find out that, the kind of, so the death of the males is predominantly after mating, then we could draw a satisfactory conclusion to our hypothesis that there might be instances of cannibalism that is concentrated after the mating rituals, and then cannibalism may be a defining factor in the mating rituals of the species. So the second one actually wanted us to verify what evolutionary advantage this whole ritual of cannibalism would have had in the population. So one of the difficulties that we kind of ensured while we encountered when we were setting up this experiment was that it was difficult to measure if there was
you're
a selective...
not audible. Is it just me, or is it with the others as well?
I am not audible.
We can hear her.
Your screen is visible, but you're not audible at all.
Oh, I am not. Adrija, she is audible.
She's
Supriyo, I think there is a problem in the line that side. Yeah.
Okay. Sorry. Continue.
Yeah, sorry.
Yeah. So one of the problems that we kind of encountered when we were setting up this experiment was that the males, if we have drawn a satisfactory conclusion to this hypothesis that cannibalism does actually occur after mating, then keeping the male alive after the mating was a difficulty. So what we kind of did was to measure the biomass of the female spiders and the male spiders, and regulate or monitor the food that we were going to give them. So what we did was that we had a female spider, we monitored the weight of the female spider and the weight of the male that the female copulated with, and distributed the food supply. So per gram of spider weight, we would assign, let's say, X grams of food to be given to the spider. So in case where after mating, we had the male and the female separated, we gave the male its body mass food due, and we gave the female its body mass food due. Whereas in the second setup, this is the first setup where we keep both the male and the female alive. In the second setup, we have just the female alive, but we have measured the mass of the female and the mass of the male with which it is copulated with, and we end up giving the female the food worth both of their body masses. So it's X2, which is the weight of the female, the pregnant female, and Y2, which is the weight of the male that has copulated with the female, and we wait. So after doing this setup, we kind of measure the size of the offspring that is produced, the survival time of the offsprings that are produced from both of these pregnant females in both of these setups, and see how efficient their energy utilization is, how efficient their reproductive fitness is, and kind of draw a hypothesis that if the male is actually alive in the experimental setup, then it might hoard resources and lead to faulty or maybe reduced development of the offsprings. So cannibalism can actually be for the greater good because it allows more resource allocation for the pregnant female, and will lead to greater reproductive fitness in the population. What will be our controls and our discussion? So for the first experiment, the data we collect is the number of males alive, and if it is alive after mating, or if there are instances of cannibalism before mating. In the second experiment, however, we collect the size and the fitness of the offspring with respect to the grams of food that is ingested by the mother. So whether it is from a mother that has ingested a food worth of both the male and the female, or just the male. What problems do we face? One is that we do have natural or accidental death problems in our first experimental setup. The spiders might die of causes that are not cannibalism, and we will be accounting for that, so that might act as a bias in our experiment. In the second setup, however, keeping the male alive seems to be a problem. So what we do is, since we are dividing the male, we are kind of destroying the natural competition that takes place in food between a male and a female, and we are kind of disturbing the whole resource allocation thing. So that might kind of trouble with the results, but on a very discrete one-to-one basis, it seems pretty doable. So what else can be done? So what else we could have done in the first experiment was we could have not have just 4. We could have increased the number of males to maybe 8 or 12, 10, and actually seen the effects of crowding on the female that is out here. So if there is more crowding, will it lead to greater instance of pre-cannibalism? So is there a pre-mating, because then they will be utilizing the resources that are present more. So the females might be pre-cannibalizing the males before mating. So all of those parameters can also be checked. And in the second experiment, we just monitored the offspring that are produced by the mother, and we could monitor successive generations to see if that is just common to what we do thanks to one generation, or whether it is something that is propagated to future generations too. What exactly are the proximate causes that we were supposed to deal with? We could find there are many, but we have written 2. One is the greater allocation of resources, which is a pretty obvious problem, because if there is a pregnant female that has a lot of eggs, it is a very energy expending process, and the death of the male might be greater. It might be for the greater good of the reproductive fitness of the offsprings. Whereas there also might be another reason. Maybe the regeneration time of the sperms of a male spider species might be very energy expending. So on the long run, it might be a greater energy intensive process to actually kill the male and just mate with a separate male every time. So it might save the population a lot of energy to do that, as morally biased as that sounds. But yes, this might be we do have a mathematical model, but I think we can come back to that at the end. So we go to the plant antimicrobial question. So it was shown that the phloem feeders did not have had pseudogenization of the antimicrobial genes. So we have understood that might be due to the fact that these phloem feeders that we were looking at had a niche. So the plants they were feeding on, there might be antimicrobials that were produced by the plants. So in that case, the plant since they're phloem feeders and they feed on the sap of the plant antimicrobials might be getting transferred to the phloem feeders, which might entirely obliterate the need for the phloem feeder to produce its own antimicrobial. So it was getting its steady supply of antimicrobials from the plant, and then it might be using it. So this whole niche that it was feeding on a plant that was supplying its antimicrobial must have led to the pseudogenization of the antimicrobial genes in the phloem feeder. And how do we exactly validate that experiment as we have hypothesized? We could do 3 setups. Setup 1, we just have sucrose and water without whatever the plant antimicrobial is there. The second setup, we have a plant extract from the phloem, which is the regular diet of the phloem feeder. And the setup 3 is a positive control. So we have sucrose, we have water, and we have the antimicrobial extracts from the plant. So we continue feeding this whole liquid mixture to 3 separate phloem feeder populations for a considerable period of time, because then the existing antimicrobials in the system are going to get flushed out. And then on successive microbial infection, we study which population of the phloem feeder actually survives, and if actually the plant extract which is fed plays a role in the survival of the phloem feeder and the niche specialization or the pseudogenization of the genes which has taken place in the phloem feeder. Okay, so now we move to the second question. Yes. So the hypothesis hypothesis 1 of the second question is that the meconium microbiome might come from the breast milk. So as after the baby is delivered, it spends a lot of time ingesting breast milk. First, breast milk colostrum is actually pretty enriched in microbiome, and it is kind of one of the primary areas where the baby gets its gut microbiota from. And we do have sufficient evidence to support this. The second hypothesis that we propose, it might be from the ingestion of the amniotic fluid. However, the amniotic fluid, as long as it is inside the placenta and is not during is before parturition, is sterile. So we hypothesize that it might be aggregate environmental microorganism invasion from the amniotic fluid that is leaked out from the vaginal and the vaginal microbiome that is present, and external factors. But what do? We have plotted the graph, which is the log of signal intensity and the log of the number of bacteria per unit mass of meconium for the 3 bacteria that were culturable. So one of them is Enterococcus, one of them is Lactobacillus, and one of them is the Bifidobacterium species. The 4th one, I think, was not culturable, so the data was not given. So this is what we have plotted. And the slope is then, what we do is we have taken the other table that was given with the individual data of individual babies and the concentration of Lactobacillus, Enterococcus, and Bifidobacterium. We have multiplied it with the slope that has come from these graphs for the specific bacteria and taken out an average. So we see that in the microbiota of the meconium, that is in the babies, a lactobacillum is a staggering 7530, which far outnumbers the amount of Bifidobacterium and Enterococcus that is present. So a major source of lactobacillus is actually breast milk. So that kind of validates our hypothesis 1 that breast milk might be a source of meconium microbiome. Also, we have plotted the graph, which shows the amount of bacterial count versus time. So we see that even after sufficient time interval after parturition, where the vaginal environment and the amniotic fluid and all of those factors are not present, we still have a sufficient bacterium count, which means that there is something constant and external that might be contributing to the microbiome of the meconium, which is breast milk, because the baby is breastfed after it is born. So this kind of validates our first hypothesis that breast milk might be a rich source of the microbiome that is in the gut and ultimately in the meconium. So question 2.2, yes, according to our analysis, we propose that gut microbiota actually modulates fetal neurodevelopment, specifically the thalamocortical axonogenesis. So what we see out here is that for the SPF, which is the wild type mice, the axonal volume is higher, which means that the volume of a nerve bundle is also higher, which means the number of neurons in a nerve bundle is higher, whereas the paw withdrawal threshold is lower. So for a lower threshold, we actually have a higher sensitivity. So the sensitivity is higher because the number of neurons are higher. Whereas in the mice where the maternal gut microbiota is depleted, in the offsprings of that, we see that the axonal volume is not very great, and the amount of weight that is needed to induce paw withdrawal threshold is very high as compared to the wild type mice. So this is what leads us to say that maybe yes, microbiome is the reason where which might actually develop, which might influence axonal development. Yeah, so the model we kind of propose out here to test this is that we know that X is found in only thalamocortical neurons and Y is found in all of the mirror neurons. So we see that when A is wild type and B is none, we have perfectly high expressions of both X and Y receptors. Similarly for this, whereas in this we see in the B is actually hypothalamic explants from the offsprings of GF. So we see that there is a low expression of X out here. So while A is perfectly wild type, B pulls the graph towards the left side, and we see that the aggregate amount of X which is present is not very high. Here also we see the same. However, for ABX, we see that the graph again shifts a bit towards the left because the expression of X might be, it might be interrupted. So maybe, yeah, is my time up?
That was 15 minutes mark.
Okay, continue on.
So we see that in here, and here we see observation that we perfectly know, that we have seen here. So we see that when A is antibiotic affected, the graph shifts to the right, which means again the expression of X, which is the thalamocortical neuron, is affected. And so is the same for here. When A is again antibiotic given, we see less expression of X, we see less expression of X here thanks to GF, we see a less expression of X thanks to ABX, and also we see a median expression of X thanks because there's a ABX out here. So what we basically see out here is that while X expression is affected, the expression of Y kind of remains constant. So Y might be a receptor that is present in every neuron, which is not affected by the gut microbiota of the maternal year, but X is a receptor that is affected, and we are told that the X is a receptor which is present in the thalamocortical neurons. While that kind of perfectly sums up our observation about power withdrawal threshold, because thalamocortical neurons are a very important method of transmission of sensory impulses, such as pressure and heat. So power withdrawal thresholds, as we see, wherever the thalamocortical neuron, the expression of X might be a membrane receptor, which plays a major role in sensory transmission of pressure. So we see that wherever there is a less expression of X thanks to antibiotic addition, we also see that the power withdrawal threshold kind of increases significantly, meaning that we have to put a greater amount of weight to actually induce a viable response. So question 2.2, the last question we see, as I've said, if we look at the FACS distribution, we see for any combination that the protein X changes drastically, which influences the power withdrawal threshold. The fluorescence intensity of the YP is almost unaltered, so which means that the protein X is only dependent on the maternal gut microbiota, not Y. From what we have understood, ABX is a mice that has grown up in a wild type, but then depleted of its microbiome, whereas GF represents mice that have been grown in sterile environments all throughout. So we see that maybe gut microbiota of the mother is gut microbiota of the mice also might be essential for continuing the expression of X. So it's not like a one-time process that if your mother has the gut microbiota, you'll have perfect neurons. I think the gut microbiota of the offspring also that is produced also plays an integral role in the maintenance of the thalamocortical receptor. So that is all for us, but we would like to explain the math model if there's time. Yes.
Yeah, sure.
So
you can take a couple of minutes, 3 minutes, to explain the mathematical model.
Cool. Sure. So what we have done in the math model is a very basic simulation. So we've taken that the ratio of the male and the female in the spider population, initial spider population, is 1:1, and mating is always successful, and no migration, immigration occurs. So the death rate of females is lesser than the death rate of males, obviously, because we are assuming sexual cannibalism to occur. The rate of cannibalism pre-mating is lesser than the rate of cannibalism post-mating. That is something that could be validated from the results of this experiment, that if it actually plays a major role in the rate of cannibalism post-mating is higher than pre-mating, and all of these rates are taken to be constant at all times and for all generations. So what we propose is a discrete time dynamics model, which means that generation, reproduction seems to be a continuous process, but for us to actually break it down into several generations and sum it up so that we get an idea of how this works. So what we have written is a very simple equation governing the dynamics of mating. P here is, okay, so small m is the total number of matings that have taken place. P is the mating success probability. So for in this case, since we have assumed mating to be always successful, we can assume this to be 1. And m is the number of viable males, which is in the population. C pre seems to be the cannibalism rate pre-mating. So the probability that individual is going to die before mating. So 1 minus C pre gives us the number of males that are alive till the point of mating, because C pre is the probability before cannibalism rate before mating. So that is what this term translates to. And this term 1 minus C pre obviously translates to the number of males that are alive before mating, and 1 minus C post being the cannibalism rate post mating, 1 minus C post is the probability that there will be a male that is alive even after mating. So, and the IN, what we have assumed out here, is the maximum mating quota. So after that, the female will die of natural causes. So we assume I to be bouts of reproduction where we sum over generations, and this is summed over as a power of I over various generations. So this term is basically the males of a first generation that are still alive after mating go on to the second generation, and we also account for the fact of the offsprings that are given from the first generation and go on to the second generation. So now we know that the product of fecundity, which is basically fertility in a population, and the number of matings would result in a frequency of mating per individual. So what fecundity, fertility or fecundity in a population is a function of? Obviously, it is a benefit of the sexual cannibalism that is occurring. If there is a benefit that we can we can kind of hypothesize or validate from the results of the second experiment, and the number of male offspring. So since we are assuming post-cannibalism to take place, rate of cannibalism post-mating is very high. So we have assumed that the number of males are almost equal to the number of matings that have taken place. So the benefits are now, since the math model is done for the second question in the question paper, where we have specifically taken out the male and then studied. So we have assumed that the benefits of sexual cannibalism pre-mating is almost equal to the benefits of sexual cannibalism post-mating. But obviously, if we do it in an original, in a scenario in a more natural environment, this will not be the case, because the benefits of sexual cannibalism post-mating is going to far outweigh pre-mating. In this case, this is going to become an iterative loop. So therefore, at a particular time and step, the number of per capita mating is going to be a function of the number of total matings times the fecundity. And this is, so we obtain the fecundity from here, and we obtain the total number of matings per here, and we can see the total number of per capita matings that occur. So, and if we want to study the fluctuations in the number of matings per individual, we need to simulate this equation over time and see if it actually gives out a stochastic picture that we can evaluate. So yes, that will be all. I think we will take some questions. Thank you.
Thanks, Adrija. So thank you. You guys finished well in time. So, and sorry for the interruption once again.
No, that's perfect.
So we'd now like to take some questions from the judges.
I want to make a comment first and then maybe ask a small question. The comment is more important than the question. Many of you, and especially in this presentation, you seem to implicitly assume that natural selection will do whatever is optimal for the population. This is not true. Natural selection rarely optimizes something for the population. It usually works at the level of the individual. And there may be a conflict between what is optimum for one individual and what is optimum for the whole population. So you must get into the habit of thinking from the point of view of the individual. For example, you said that in natural conditions, post-copulatory cannibalism will always be more beneficial than pre-copulatory. From whose point of view? There are lots of males in the population. Then for a given female, she might as well eat 5 males before copulating, get all the energy she needs, and then after copulation, she may or may not cannibalize at all. From her point of view, it's not obvious that post-copulatory cannibalism is more beneficial than pre. If you're looking from the population point of view, yes. So you must get into the habit of thinking from an individual's point of view. In fact, from individual female's point of view, individual male's point of view, what is optimum for them? So in
Kamala, sir. So yes, I understand what you're talking about. I think this was the reason why we had included the crowding in the discussion. So maybe if we have a lot of crowding, there will be instances of pre-cannibalization that will occur. So that was an assumption that we had included. But yes, we had forgotten to include the male's perspective. Crowding would influence only what the female does, but it would not kind of influence what. We did not include.
Female's perspective is one aspect, but more generally, natural selection will not optimize what is good for the population. It will optimize what is good for the individual. So as I said, a given female, if she is capable of consuming 4 males before copulation, she will do so, unmindful of whether the population will grow or not. She doesn't care whether the population will grow or not. She is only worried about her fitness. And that's the way you should look at. So one way to think about this is of course difficult to do in an experiment, but in a model, you should start thinking about how you will contrast these two. So what is being optimized in one model is what is good for the population, in another model is what is good for the individual. So that will help you to compare these two.
And then maybe we could input the outputs of the individual model into the population model and accordingly do it.
But in general, this is a standard problem in understanding.
Okay, sir. I will keep this in mind.
Just saying this is something that you should keep in mind. But my question was the beginning of your experiment. Can you quickly take me to the first 1 or 2 slides of your presentation?
Yes, sir.
Just to refresh my memory. I enjoyed your presentation, by the way. I'm not criticizing it. Some clarification. All of you guys present with such great confidence. I wish my PhD students had that kind of confidence. I'm impressed with the level of everybody's presentation.
So, are you looking for this slide? Should I go forward?
Next one. So you had experiment 1 and experiment 2, right? So in experiment 1, you were going to infer that there is post-copulatory cannibalism if the males disappear.
Yes,
right? Sir. Now, of course, it's possible that males just die, as you also said later on, from some completely different causes.
Yes, that is exactly why we have tagged the males out here.
Accident and so on. But even more fundamental, let us say the life history strategy of the male is to mate and die. His life ends as soon as he mates.
Okay, sir.
With or without any cannibalism. That is entirely possible, and it happens in some species. Males are very short-lived, they donate sperm and they just die because their lifespan is over. And there is no no need for cannibalism for them to die. So in order to distinguish between those two options, you may have to do something more than just count the number of males remaining alive.
Sir, in that case, so if the males kind of die just after mating and it is not an instance of cannibalism, then technically the resource allocation should not be biased. So if we have an instance of cannibalism, then the resource of the male would be utilized by the female. But if there is if the males voluntarily die after mating, then the female would be allowed to feed only on her resources and not on the male's resources. So could we use that as a defining factor to distinguish between whether cannibalism actually occurs or whether it's just the life cycle of a male to die?
That's one way to go, but unfortunately real life is more complicated. So the allocation of resources depends on so many other things, not just this. But you're in the right direction. But one must worry about. So I was thinking that none of you actually considered the possibility of putting a video camera there and actually seeing what happens. Isn't it a lot of fun?
So this is what we did, sir. We tagged the monitors and we were I mean, what we meant by tagging out here was actually monitoring it via video camera to see if they actually died after mating or not.
Tagging usually means you put a spot of paint on it and then so that you can recognize which is the animal.
Yes.
Now tagging that word is not used. So if you actually video some of it, you can actually see what is happening. Behavior is very interesting to observe, and counting is okay, but actual observation is very interesting. So nowadays it's very easy to put up simple cameras and get a few hours of video and then analyze those videos. So I was just a suggestion, but I enjoyed everything. Thank you.
Thank you, sir.
Adrija, I'll just ask you, very nice presentation. Just
Thank you, ma'am.
Stay on the slide. Can you go to this experimental setup too? The next slide, I think. This one. So you be here. So in this particular setup, you mentioned that since the males are not alive after mating. Now, this word alive can be of two things, that the males are killed and the males are killed and have undergone cannibalism. Now, the word cannibalism by definition means that species is feeding on the same species. That means the female spiders are feeding on the male spiders, and the cannibalism, that is the eating, needs to be preceded by the killing, right? So the killing plus eating is cannibalism.
Yes,
So in this case, when you say since the males are not alive after mating, you essentially means that it is cannibalized, right?
yes,
or do you mean that it is just killed? They are not alive. What do you mean here by alive? That means it is cannibalized.
yes, ma'am. I mean it's cannibalized. It's killed and eaten.
It's killed and eaten, right? So now if
Yes.
it's killed and eaten, now the food portion of the female plus the killed and eaten male plus the food of the male. So there are
Yes.
3 components here, right?
Yes, yes, right.
Yeah?
Right. I think we have forgotten to account for one factor.
Yeah. You got it, na? Here?
Yes,
Am I
yes,
right
yes,
in
ma'am.
saying that?
Yes, ma'am, you are absolutely
Okay.
right in saying that. We have
Right.
forgotten to account for.
Yes, so that is one thing. Second, if you can go back to your tagging experiment, it's just I wanted to know for my own understanding. Yeah, here. So now here you are saying that you have set up one female with 4 males and you're going to monitor, etc., etc.
Yes.
I am just trying to understand that, in this entire story, we are only trying to see the male as a source of mating, as a giver of progeny, and as a food, both.
Yes.
Now, we all know that the spider have other food sources also, right? Like suppose insects and grasshoppers and crickets, etc. So what if now you put the other food source in this particular situation and then ask a question pre and post, that do they prefer the other food source over the so-called male? And also, why only male? Even the female spiders, pre-copulation and maybe post-copulation is the male is now killed. Now the killing followed by cannibalism. This is 2 totally different, activities that are taking place. So is it possible that if you put another food source here, say along with 2 male spider, you put a grasshopper or a cricket, what will be the situation, or how will your scenario look like?
Ma'am, in this case, we just have 4 spiders, so I don't think the case of crowding actually occurs where, I mean, this is just all hypothesis because we don't have enough data to back it up. There might not be enough case of crowding for the female spiders to actually go and eat the male spiders. That takes care of the pre-cannibalization part. If we put a grasshopper in here, I preferentially think that reproduction is also it's one of the major incidences of what drives evolution. So given a choice between food and like a food that is supposed to be a food and a food that could be preferentially a mate, a spider should choose the food that is supposed to be a food. I mean, that is something that I think that should occur. And between 2 female spiders, they're given to be given to, like the female spiders are they said that the female spiders are significantly bigger than the males. So if there are 2 similarly sized females, I think if there is a fight between the 2 that results in cannibalism, then again that would be eliminating that would be higher reproductive fitness if we look at it that.
Can you quickly go to your phloem slide?
Yes.
1.2 question. Yeah. Here you have very beautifully, I really liked your view that the plant antimicrobials may be present in the phloem, and that is how when they suck the sap, it gets inside. Yeah, then just move forward one slide. Okay, now in this particular experiment, you are putting up again a very nice experiment of sucrose plus water, plant extract, and now then you are trying to understand that really whether the phloem is containing your antimicrobial peptide.
Yes, ma'am.
Now, how will you do this experiment? I was not clear.
Okay, ma'am. This is, I think we did not exactly go into the how of the experiment, more concentrated on how.
How will you prove that your plant extract actually contains the antimicrobial?
So, ma'am, what we assume out here in setup 1, setup 2, and setup 3, we have individual populations of phloem feeders that are kept separately. One of them is fed on a diet of sucrose and water. Second is fed on plant sub that it normally feeds on. And the third, we have a positive control, which is sucrose and water and antimicrobials that we have extracted from the plant. Now, we already know that these three setups contain phloem feeders that have been feeding on plant sap for a really long period of time. So there might be the antimicrobials that are there in their system, which came from the previous exposure to plant sap before they were caught and brought for the experiment. So
no,
we
but how do
continue
you
feeding.
prove that they have the antimicrobial activity? That was the question.
So we infect them after a significant period of time of feeding with these extracts. For the setup, we infect them with microbes that is supposed to be there. Like we could analyze the plant extract and see what kind of microbe it defends for, and then infect the phloem feeders with that particular microbe with which the plant extract might provide protection against, and see which setup actually dies out.
Okay,
So,
good.
and then monitor the setup.
Yeah, the third question is vaginal passage contains more lactobacilli or your breast milk?
Ma'am, it was shown explicitly that breast milk, especially the colostrum, is a very rich source of where the microbiome of the gut of a baby comes from. So meconium could. So
no, but which one contains more, your vaginal passage or the breast milk?
Yes,
okay, if you don't know the answer and if you would like to put up an experiment, no, but if you would like to put up an experiment, can you design?
Just a second, I will just think about it a little bit. We have to put up an experiment. Yes, ma'am, we could do so. We could do so based on the fact that we could have two independent samples of babies. One of them were in just breastfed, and the other sample of the babies that were not breastfed, that were just fed exclusively from maybe packaged milk that was sterile and did not have any microbiome. And then the meconium could be monitored. So if there was a lot of so if there's like a lot of microbiome in the meconium of the first sample that was breastfed after a significant period of time, because then let's take a time period of maybe a week or something where they're breastfed with just breast milk and the second sample is fed with just artificial milk, condensed milk maybe. So then there's a significant period of time after which the baby has come out of the vaginal canal. So any sort of microbiome that would have come, if it comes from the vaginal canal, then there will be like significant microbiome in the second sample, whereas if it is there in the first sample, then it might exclusively come from the breast.
Okay, thank you.
Thank you, ma'am.
Any other questions from the judges? Yes, Professor.
Yeah, I don't have a question, but I would like to just compliment that it was a very good presentation. Whatever I wanted to ask, whatever I wanted to ask, Deepshika has more or less covered it. But just one simple thing is that how you just think about it. I mean, how you will show that antimicrobial peptides are deterrent to, are trade-off to what we call a lifespan, right?
I'm sorry, sir. I don't think I've got your question.
how you will be able to show that antimicrobial peptides have a trade-off to lifespan or the physiology of the organism?
Okay, sir. So that I think what we could do is just look at and monitor lifespans of phloem feeders that do not have pseudogenized antimicrobial genes from close relatives, and see and monitor their lifespans to see if this pseudogenization actually affects the lifespan, because producing antimicrobials is also a very energy extensive process, and if minimization of that while getting it from the plant actually saves on the lifespan of the insect, then it should show like a significant monitoring of a lot of samples of this phloem feeder and its close relatives which do not have pseudogenized genes should show a significant deviation of lifespan. So I think that could be a way of checking if pseudogenization actually affects the lifespan.
Okay, right. Thank you.
Thank you, sir.
Thanks, Team IISER Kolkata. We really do not have any time for any further questions from the floor, so we'll end it here for you. Thanks a lot. So now we'll go to the last presentation of today's session. The last presentation will be given by Parth Chandak from IISER Kolkata. Parth, are you here?
Yeah, can you see my screen?
Yeah, I can see your screen.
Okay, cool.
And we can hear you as
what
well.
About now?
Yeah, it's your slides are visible.
Okay.
Yeah, so your time starts now.
Yeah, hello, I'm Parth. I'll begin with the first part, Ecologist's Wonderland. So for the first experiment, the null hypothesis is that there is no post-mating cannibalism, and alternate hypothesis is that the males are cannibalized by the females after mating, or that post-mating cannibalism is present. We set up two enclosure types in a greenhouse or an ecology lab or field station close to the natural habitat of the spiders to control the environmental variation, possible environmental variations. The prey the spiders are fed are kept similar in number and species composition to control variation in nutrition. The enclosures are made in such a way that no emigration or immigration of individuals of the spider species is allowed, like through provision of small holes in the enclosure only. We keep checking and removing predators of the spider species if they get inside, and we randomly assign the individuals to the enclosures to eliminate any natural bias. In the first enclosure type, which is the control, we put females and males and prey of a spider species not known to show sexual cannibalism. In the second enclosure type, which is the experimental, we put females, males, and the prey of the species we are studying along with the prey. We observe all matings and all events of cannibalism in the enclosures. We normalize the number of cannibalism events post-mating to the number of matings that occur in the enclosures. If we observe that there is significantly higher number of cannibalism events post-mating in the new species, in comparison to the species not known to show cannibalism post-mating, it would mean that sexual cannibalism post-mating is present in the new species. How common it is in the species is something that will depend on what cutoff values we set for the frequency of cannibalism post-mating. If we find that sexual cannibalism post-mating is present, we need to find if it leads to greater reproductive success. The null hypothesis in this case would be that it does not lead to an increase in reproductive success, and the alternate hypothesis will be that it leads to an increase in reproductive success. All right, we again set up similar enclosures. So basically with similar methods of control of the environmental factors as before. Now in both enclosures, we put females, males, and prey. In the control enclosures, we allow mating to happen, but do not allow post-mating cannibalism. This can be done through physically pulling apart the male from the female as soon as the cannibalism is attempted post-mating. In the experimental enclosure, we allow mating to happen and cannibalism post-mating, if it occurs. Reproductive success depends on the number of eggs produced, their size, and the offspring survival to sexual maturity. So if in the experimental group, we find that females that cannibalize the males produce higher number of eggs, larger in size, and or the spiderlings show better survivorship to sexual maturity in comparison to the control group females, it would mean that there is greater reproductive success due to mating post-cannibalism. Now, here we can also study another thing, which is the male destined to die naturally after the mating? So once we separate the males from the females in the experiment above, we observe the male survival. If the majority of the males die, then that would mean that they are naturally destined to die. But if they don't, then it would mean otherwise. So what could be the proximate causes for greater reproductive success? It could be that the male is a source of nutrition to the females, but this would depend on its size relative to the female. Also, recently it was shown that females of a species of spider, Dolomides tenebrosus, when given a cricket in place of a male to devour post-mating, do not lay more eggs or have higher offspring survival. This means that the males may not be important with respect to nutrition. Instead, they could be important as a source of a chemical that causes a greater number of eggs to be produced in the female body and better spiderling survival. Also, eating a male may also introduce more sperm in the female body that could lead to more fertilization of eggs in the female body. Finally, it could be due to elimination of competition for resources and like an attempt to eliminate competition for resources, particularly when resources in the environment are scarce. This may not be true for the species that mates in the monsoon, as monsoon are generally good time for prey availability. All right. So the pseudogenization of genes in phloem feeders. In my opinion, the pseudogenization could be due to two reasons. The plant phloem sap is a source of antimicrobial compounds that the phloem feeder can accumulate. An indirect analogy could be the monarch butterfly that accumulates toxins in the caterpillar stage and retains it through the adult stage. Another reason could be the evolution of an ability to detect and avoid pathogenic bacteria and other pathogens. So, for example, pea aphids can use vision in presence of UV light to detect the presence of pathogenic bacteria. The pathogenic bacteria have a molecule called pyoverdine that fluoresces in UV light, and hence it can subsequently avoid them. All right. So the experiment to test the kind of selection that made them evolve pseudogenes, we could use two sets of phloem feeders, one that feed on phloem sap and another fed on sucrose solution. Both these sets are first kept in sterile conditions and then exposed to pathogens for some time known to infect these phloem feeders. Survival is measured in both sets. If you observe significant survival in a set fed on sucrose solution, it would mean that there is balancing selection due to which multiple genotypes are surviving in the population. If you observe no survival in the set fed on sucrose solution, it would mean that the genotypes in the population is one with pseudogenes and no anti-pathogenic resistance is present. We can also do an experiment in which we use a close relative of the phloem feeder that does not show pseudogenes. We can feed individuals of that species on a diet that has antimicrobial compounds in it and observe if resistance gene evolved to become pseudogenes. But this study would require evolutionary timescales that may make it not so feasible depending on the species life cycle, the species that has been chosen. All right, neonate gut microbiota acquisition and neurodevelopment. So according to the given data, the newborns have predominantly Lactobacillus, Enterococcus, and Bacteroides prevotella family species, although very low in number. Although I could not put cells per gram values for the fluorescence in the table, the cells per gram in the table are of the order of 10 to power 3 to 10 to the power 10 to power 2 to 10 to power 3 cells per gram. Lactobacillus, Prevotella, and Enterococcus are known to be present in female genital tract and have been shown to be transferred either via the amniotic fluid or from the membranes of the birth canal during vaginal delivery. But as we can see in the majority of the babies, either no cells of the bacterial species are present or only few of them are present at birth. The total number of bacteria show logarithmic increase with increasing Meconium interval. This means that after the birth, the neonate is rapidly developing gut microbiota. An important thing to note here is that the baby is being breastfed by the mother after birth. So the genera of bacteria that have been isolated from breast milk are Lactobacillus, Enterococcus, Bifidobacterium, and Streptococcus, to name a few. As some of these are the ones that are being studied in this here using fluorescence in situ hybridization. It can be said that the main mode of acquisition of gut microbiota is from the breast milk and the skin through physical contact with the skin of the mother. Acquisition from vagina or before the childbirth may be happening, but it is not significant and or not common in neonates. Another hypothesis can be that the baby is acquiring very low amounts of bacteria through amniotic fluid, but the acquisition does not lead to growth. Instead, it leads to only a transfer of the neonate gut, and the bacteria start growing once the neonate is born and is exposed to the external environment, which could explain why the initial cells per gram values are low, but later they rise exponentially, like in this graph. All right, fetal neurodevelopment. Data for axonal volume and paw withdrawal threshold was provided for 3 conditions. SPF, which means conventionally colonized specific pathogen-free wild type, GF, which stands for germ-free, and ABX, which stands for antibiotic treatment from conception through mid-gestational period. So the axonal volume in SPF was significantly higher than GF and ABX. Also, axonal volume in GF is significantly higher than that in ABX. This would mean that early colonization is crucial for brain axonal development, and colonization beginning after mid-gestation cannot rescue the loss in axonal development. Also in nociception, the paw withdrawal SPF, the paw withdrawal of SPF has much lower threshold than GF. ABX also has a lower threshold than GF, but greater than SPF. This can mean that colonization is important to develop nociception, which is part of the peripheral nervous system. And with colonization, even after mid-gestation, we can develop nociception. Finally, it is given that X is an axonal membrane marker. Y can be a housekeeping gene or a constitutively expressed gene, which is used as a control to keep a check on protein expression levels of the neuronal cells. So higher the presence of X, higher will be the neurodevelopmental state. We can see that SPF individuals have higher neurodevelopmental levels than ABX and GF in order. So this would imply that colonization is important for early neurodevelopment. Do I have time for explanation of the mathematical model?
Yes, sure. Go ahead. You have time.
Yeah. Okay. So coming back to the cannibalization phenomena, so my mathematical model is based on the assumptions that there is no pre-mating cannibalization events. All matings are successful. There's a natural limit to the number of times a male can mate, which is denoted as K, and there is no immigration or emigration happening in the population being studied. So we define here mating probability as P submating, or simply P, the viable males as M, the number of viable males as M, and probability of post-mating cannibalization as C. So the total number of matings in the population will be P times M for the first mating. Then if the male survives, then it would be P times M times 1 minus C, and then a geometric progression. Since there is no limit to the number of times a male can naturally mate, since there is a limit to the number of times a male can naturally mate, which is K, the total number of matings possible is PM plus this expression here, which can be condensed into P times M into 1 plus sigma 1 minus C to the power n, where n goes from 0 to K minus 1. The total number of matings per individual will be just the above expression divided by M. If there was a chance of pre-mating cannibalization too, which is denoted as C prime here, then the total number of matings per individual will be equal to P times 1 minus C prime plus the above expression with 1 minus C prime multiplied by 1 minus C, which is basically the likelihood of the survival of the male. So we can also find out the number of offsprings that will be produced per individual. Offsprings produced when no cannibalization happens. Let's say it is denoted as N0. If the female cannibalizes the male, she gains some reproductive success according to the question. Let us call this advantage as A. When cannibalization happens, the number of offsprings produced is N0 times 1A. So the total number of offsprings produced per individual will be the expression for matings per individual times N0 1 plus A. Now the C in case, so we can define A as a function of C. Like we can say that, let's say if there is a benefit to the female by cannibalizing the male post-mating, then we can define it as, for simplest possible expression, we can define it as benefit times the cannibalization rate. So it would be 1 plus, so it becomes B times C. So it would be N0 1 plus B times C. And if the male has advantage in not getting cannibalized and it tries to attempt to not get cannibalized, then we can also associate C prime and C or with a dividing factor for the males. So we can denote it as so basically we can add a dividing factor here, and that will basically incorporate the advantage for the female to cannibalize and the male to evade cannibalism. So that is that. Thank you.
Thanks, Parth, for the presentation. So we'll now like to have some questions from the judges.
I want to ask you something about the early part of your presentation. If you can
Yes,
go
sir.
to the slide where you listed the multiple hypotheses for the advantage of cannibalism. Can you take me to that slide? One of them was,
Proximate causes for greater reproductive success.
this one. Yeah.
Yeah.
So now, how can you think of experiment that will help you to discriminate between these hypotheses?
So one is.
Do you think this hypothesis is reasonable that eating the male may induce more sperm into the female body? Do you think this is a reasonable hypothesis?
Third, I mean, it was one of the hypotheses that came in my mind. I did not think of planning an experiment.
No, but don't you think sperm will get digested in the heat rather than go and fertilize the egg?
Yes, sir, definitely, but
possibility, no? But in any case, we can always put on the table some hypothesis which will not stand up to scrutiny, because we need something that will not stand up to scrutiny. So can you think of how you will distinguish between these hypotheses?
So I think so for the last hypothesis, I am claiming that since the female is mating in monsoon, where prey availability should not be a problem. So I think elimination of competition for resources would be a better hypothesis if we,
I don't think that would be entirely correct, because you can't just say because it's the monsoon there is no problem of prey availability. That can't be. You have to compare monsoon with some other time, or you experimentally remove the food, no? Because you think there's a lot of food in monsoon. The spider may not think so. She may find it much easier to lap up the male who is just next to her than to go and look for some food. So unless you do some experiment, you really can't say because mating happens in the monsoon, this cannot be a valid hypothesis. You will have to do something more.
So what we can do is basically have different densities or like different numbers of individuals in the cages, I mean the enclosures, and see how the cannibalization frequency post-mating is changing.
Yes.
If we have lower number of individuals, so if the elimination for competition for resources is true, then we should see less amount of cannibalization post-mating, and vice versa for higher.
And for the last one where you talk eliminating competition, can you think of an experiment? The male is the purpose of eating the male is to eliminate competition, right? That's what you're saying.
This is all right. So I claim this with respect to the basically the individual itself, not as a female or as a male. Eliminating competition for resources. I mean, basically food resources in the sense.
Yes.
Yeah. So
think of what will support this hypothesis or what will disprove this hypothesis.
I mean, similar to the experiment I described before, it could be like you can maybe vary the number of individuals of a particular sex and see if there is competition.
But if a female spider eats a male spider in order to reduce competition for scarce resources, the benefit of that will go to everybody, not just to her. Right?
Yes, sir.
So then it becomes an act of altruism. I'm eating my male so that all of you benefit. I'm getting more food. Right?
Exactly. Yeah.
Let us find out that things are fairly complicated. So you'll have to think about how to test these different hypotheses. There are always multiple factors influencing everything, not just. And that's why at least lab experiments will try to will help make it somewhat possible to isolate individual factors. In the field, everything is operating at the same time.
Exactly, sir.
Yeah. So in spite of some cost of lab experiment, a combination of lab and field experiment is always the optimum choice. Yeah, just about it, Yeah, very good. Thank you.
Yeah, Parth, very nice presentation. Parth, can you go to your phloem story?
This one?
Phloem, yeah. Pseudogenization of the genes in the phloem feeders. Now, here you have written the second point is the detection and avoidance of pathogenic bacteria.
Yes, ma'am.
Intake and accumulation of antimicrobial, that is from the plant itself. That is fine. And the detection and avoidance of the pathogenic bacteria. If you would like to indeed test this hypothesis, can you tell me how would you like to put up an experiment? And very interestingly, you have given an example. P. aphids can use vision in presence of UV to detect and avoid pathogenic bacteria. Now, pyoverdine is not the bacteria. No,
ma'am. I mean, pyoverdine is the molecule that fluoresces.
Yeah. And pyocyanine and pyoverdine, these are the two important molecules that are produced by Pseudomonas aeruginosa. Yeah. So this is the pathogenic bacteria is Pseudomonas aeruginosa, and the pyoverdine is so-called the toxin which is produced. Yeah. Now, detection and avoidance of pathogenic bacteria, this particular concept, can you tell me how would you prove that in your system?
So what we can do is assuming the premises of seeing and detecting it on, say, a surface. So what we can do is basically coat surfaces with pathogenic bacteria, pathogens that affect these phloem feeders, and see if it can actually see if it can actually distinguish between distinguish with more than chance rate the difference between basically the surface that has not been coated with pathogens and the surface that has been.
No, so, if your surface is coated, suppose if your plant surface is coated with the pathogen and your phloem feeders are feeding on the sap, they can take the bacteria along with the sap, right? So how that detection will happen, how that avoidance will happen? During the feeding stage, you mean that the avoidance will happen during the feeding stage?
I mean to say here that basically the place where the phloem feeders are actually feeding, let's say they have a plant and they determine to feed on it. So the point where they are going to impinge, that will be determined according to basically the density of pathogens or the presence of pathogens around that position. That is what I'm trying to.
How? I mean,
so that is through. So that is through UV light. I mean, in P. aphids, it has been seen that UV light, basically under UV light, the bacteria will fluoresce. So if you see that there is fluorescence on one part of the plant, so that means you will just eliminate it as a point of impinging.
And if there is no UV light in that case, suppose there is no such fluorescence that can be detected.
So, I mean, it will depend on what kind of detection and avoidance mechanism they develop.
Okay, that's fine. That's fine. You are fine, Parth. Parth, in your mathematical model, I was just curious if you can just show your mathematical model once. In that particular model, of course, you have built in many of these conditions. I was wondering whether you have also built in a condition where the male is killed but not cannibalized.
Okay. The male is killed but not
Killed.
cannibalized.
Yes.
Can
that happen?
Okay, so I mean, I can add a term here.
Yeah, I'm just.
So along with 1 minus C, I can just simply put a term here if it is actually that simple. I mean, I don't know if it will actually work. I mean, it will actually replicate what happens in a natural population. But as far as I can think about this, I think 1 minus C times, 1 minus C minus the probability that it will only be killed and not cannibalized.
But not cannibalized. Okay.
Yeah.
Thank
so
you.
Then, yes, ma'am. Thank you.
Yeah.
I think you can see immediately that this suggestion that Professor Deepshika has made relates to your hypothesis of eliminating competition. Right?
Yes, sir. Yes, sir. Exactly.
This is how you should see the connection. Because if all you want to do is eliminate competition, just kill, why eat? Right? Whereas if you eat, that is something more than eliminating competition. So you can immediately see that these can help you to distinguish between these different hypotheses.
Yes, sir.
Thank you, Professor.
Sure.
Thank you, sir.
Yeah.
Part in your phloems that pseudogenization.
Yes, sir.
Is it possible that your neutral selection can happen if you have multiple copies of the multiple gene copies, right? By duplications, right? Yeah, and when some of the losing 1 or 2 copies, I mean, it will not harm the organism at all, no? Is that possible?
Yes, sir. Yes, sir, it is possible, but the way I've interpreted this question is basically that pseudogenization of the gene itself. Irrespective of the number of copies.
Yes.
But in your case, you're asking that is it possible that neutral selection will lead to
no.
Basically,
I'm just telling that how whether your neutral selection can be explained by having duplicated genes things, and only 1 copy is enough just to do the thing. So the other genes can duplicated genes, right? They can pseudogenize, right?
Yes, sir. I mean, if one can do, then others should pseudogenize.
Yeah. So this has not been considered by any of other your presented before you also, right? And
yes,
that happened in the many genes actually in higher animals where you have multiple copies of these genes. Many of the things are non-functional because they form kind of redundant, right? Okay.
Yes, sir. There is, as far as I know, there is a phenomena called accordion expansion and basically compression, wherein for selection you basically create lots of copies of a gene, and once the more active gene has evolved, then you basically lose all the others and keep that gene.
Right. That's correct. Thank you, Parth. Good presentation.
Thank you.
Thank you, judges. So we are really short of time now, so we won't take any more questions from the floor. So we'll now go ahead with tabulation of the results and declaration of the results. So before that, I would request the judges to please meet on the judges channel. The participants are requested to continue in this meeting because we'll be announcing the results shortly. And till the time we are tabulating the results, the volunteers will be here. You can chat with them and relax a little after this hectic presentation schedule.
So should we leave this and you will send us an invitation for the other one?
Yes, sir. Yes, sir. I'll call you from that meeting. Yeah.
Okay.
Okay. Hi, guys. We'll just like to ask you about your experience of BioBlitz and your review, suggestions, anything you want to tell us about this entire journey, the prelims, the finals, everything that happened. So maybe we can go serially. Suhas, Mahima, Ananya, your team. Hello, am I audible?
Okay, maybe they are not there at this moment. Maybe anyone else could speak up. You can speak anything, like whatever, if whether you like this event, dislike this event, whatever
Yeah,
feedback you
the
want
team,
to give.
the team from presidency, Ashmita, Srijoni, and Onnisha.
Yeah, I'm Srijani. I really like the experience. It was a great learning opportunity, and the questions of all the rounds were very inspiring and thought-provoking. And so, yeah, I appreciate it and liked the experience.
Any suggestions for us?
Any questions can be improved, or the format, any such thing?
I don't think we have any suggestions. Actually, I love the analytical aspect. Means usually we study the theory and don't get to analyze stuff like this. So this was very enjoyable.
That felt really good. Thank you so much. The team from Pragati Public School, Priyanshu, Dhiren, Anshul.
So we really enjoyed the event and it was really very fun. We came to know about various new things and yes, it was a very good experience and the mathematical model thing was really innovative and I really liked that question. And even that idea of framing question from research papers was very innovative and net I really enjoyed this.
Okay, like anyone from your team, like Anshul or Priyanshu, do you want to add to that?
No, it was overall good.
Okay, we are pleased to hear that. Okay, the team from Isaac Kolkata, comprising Sharman, Audrija, and Tisha.
Hello, am I audible?
Yeah, you are audible.
Yeah, so I guess like BioBlitz for me was a very interesting concept. Like we have also participated in other competitions, but this was the first time we were able to look at every question in another way. My other finalists also said it was extremely thought and also analytical, and we actually enjoyed a lot. Like we learned a lot. We had the opportunity to learn a lot because almost like all the questions, if we look at all the questions together, they were comprising of different branches of biology, not just ecology or not just like molecular biology. It comprised of almost everything. So it was really very interesting. And for suggestions, frankly speaking, I liked it very much, and I personally have no suggestions because I felt it was really good. So like if any other person wants to comment something on this.
Yeah, so I would like to add in this one thing that at the beginning of the prelims round, what the organizers have told that no matter what team will qualify in the successive rounds, all will enjoy at that and will definitely learn something out of that. So I literally personally enjoyed all this thing and learned a lot while accessing these questions. Prelims one was really challenging, and finals is also really challenging for me. So, and I enjoyed a lot.
Yeah, I would just like to add that, yep, I agree with my teammates. And I mean, it's kind of a breath of fresh air when you actually see that biology is finally not about memorizations, but actually applying whatever you've learned and analyzing it. So it's really nice. Just one suggestion, maybe you could just reduce the time because it's very fatiguing when you go towards the end. So yeah, but other than that, I thoroughly enjoyed the questions and the experience and everything else too. Thank you.
Okay, that review was really heartwarming. And okay, yeah, we have noted.
Sorry.
Maybe. So
The
Dhanva, do you want to
timings
say
were
something?
very.
No, I said
Okay,
thank
yeah.
you for the feedback.
Yeah, okay, yeah, we'll try next time that we don't take it so long and complete it
Yeah,
well
I mean
within.
2:30 at night, your brains freeze at that point.
Yeah, I understand. I totally understand.
I think when it was like other events, like BioBlitz is the debut year, but earlier like other events, I think used to be experimental rounds. Like that used to be more fun and more energetic than this. Like sitting in front of a screen does get tiring. I agree. But
Yep,
actually,
but other than that, the whole experience is really rewarding. So I guess it balances out.
yeah. Like even as a volunteer, I think I learned a lot just by framing the questions and reading the answers.
But I personally think that although the time is bit uh, more, but as a researcher in this field, we need this pressure in our mind that the timing, but this timing that was given is not fully is beneficial at a point, at some point, because it trains our mind also.
Well, yeah, that's true to an extent. But Adrija had a good point that 2:30 a.m. is sort of a brain freeze moment. So yeah, okay, yeah, that
Yeah,
we get that.
that's no.
We get that. It's cool, cool. Thank you so much. Okay, Swapnomoy from IACS. Swapnomoy, are you with us? Okay, maybe not. Parth from IISER Kolkata. Okay, no.
Yeah.
Okay, yeah. Hi.
Hi. Yeah, so it was like really interesting from the prelims 1, 2, and the finals. I'll say I've learned a lot of things, learned how to design experiments, controls, and stuff like that. Yeah, I think it was really cool, even though it was the debut year of BioBlitz. I think I have a lot of takeaways from here. Even if I don't win, I mean, I'll be happy that I have learned so many things.
Okay, thank you so much, Parth. Do you have any suggestions for us? Like it's the debut year, so we are looking forward to suggestions so that we can improve the next time.
I think it was, I don't know, I don't have any suggestions. It was really well organized. No, I don't think I have any suggestion. I was
Okay,
more or less satisfied with it.
Okay, cool, super cool. Okay, Anurag from Yogiraj School.
Hi.
Hi, Anurag.
So I did have a lot of fun. It was kind of hectic, but I think I enjoyed that to an extent, in the sense it was nice to sort of be in that zone for like 2 or 3 days. So it was all in all like a fun experience for me. One possible suggestion I might add is for like the round, the review paper round, right? There was this question on this like pathfinding and DNA, right? So like the question was pretty like sort of cool, right? In the sense that what it was referencing was very interesting and very new. But I felt like the question effectively needed the person to literally look up the original paper to even like just like to understand what is going on, because like I didn't feel like it was something that was realistically somebody at our level, or even just like a person giving an exam in general, whatever be their level, would be able to reconstruct in like the time given to us, or just like the format it was presented to us. I mean, it was like a nice question, like once you found like the actual study in question, right, like the DNA Adelman study, it was pretty like mind-blowing stuff, but I just felt like the premise of the question could have been better defined. So yeah, if that makes sense.
It makes sense. That question was sort of, we collected all the questions and we saved it for the finals because it sort of looked like out of the box, but yeah, we realized that it might have got a bit overload if not properly defined. Okay, yeah, we have taken that into account. Thanks. Okay. Are Suhas, Mahima, and Ananya here with us?
Yeah, yes. So, like, this was actually very interesting, and I found like this was way better than other competitions. For example, I have participated in like many other such competitions, but like this was way cooler than most of the other competitions, and like I actually learned a lot in this. Most of the things were not related to my field, but like I learned a lot, and like this was pretty cool. I like really loved it.
Mahima, Ananya, would you like to say something? Add to it, suggestions, anything? Okay,
Yeah, can you
cool.
hear me?
Yeah, we can hear you.
Yeah, so it was very good. Like the research papers that were given were very good. It was hard to select one because all of them were very interesting at the very first place, and we had a lot of fun as a team discussing all the questions. Yeah, it was a very good exercise for all of us. So it was very well organized. Thank you.
Okay, thank you so much.
Yeah, so if we are we done with asking everyone for the feedbacks and suggestions?
Yes, Om, we are.
Huh. So maybe if you don't mind, among the 4 teams that were selected, only one of them was a school team, and comprising of Priyanshu, Thiren, and Anshul. And among the 3 pre-Pravega participants who were present today also, one of them was a school participant. So can you share your interest in biology and how you got interested, and what particularly interests you, and what would you like to do in the future?
Okay, so yes, I can go first, if that's okay with the
Yeah, Anurag,
team.
yeah, you can.
Okay, awesome. So my interest in biology primarily comes from preparing for the biology Olympiad in like 2019 and 2020. So like I gave the exam twice. So that's primarily where my basic interest in biology comes from. Beyond that, in terms of my interests, I'm really into ecology, ethology, and I mean it's not something I have usually heard people like, but or like talk about much in like these spaces, but philosophy of science as well is something that particularly interests me. So yeah, my plan is to like again get a BS, MS, or a BS from like a NICER or IISc in the future. I'm in 12th grade currently, so I'll if like COVID restrictions allow, I'd probably be attending college next year. So yeah, it's that.
Okay, great to hear that. Probably you will be one of our juniors, or one of the people at ICER. So maybe those junior of someone who is participating right now. So the others, would you like to share?
Well, yes, I am Dhiren and I am from 12th class, and my interest in biology was primarily because of I was interested in cancer, and then I started reading things related to it, and then I went into genetics and I read more about it, and and then I just kept reading, and now I am like quite confused, but yes, I am in pretty much interested in all in a lot of fields of biology, and I really like biology. In fact, I even like evolution, and but as
Why don't you put on your video when you speak, sir? I can know who is speaking.
There, it's always good to be confused at this stage of your education. Actually, it is always good to get confused, because if you are very clear from now, then I think you will be super smart and super intelligent person. I mean, you could be okay, but I think, I mean, there is no harm. We all start with confusion, I think, right? That is how we keep ourselves motivated, because any field, if you take it today, any field, there will be confusion and something which we don't understand. That is where we try to, solve the problem and we get excited. But remember, whether it is in cancer or genetics and things, what we understand today, tomorrow somebody will say that that is actually wrong or right or something, some kind of variant. So the science is self-correcting thing. So just get yourself confused all the time, but don't give up. Don't give up. That is the most important thing. You keep yourself motivated. I mean, Gadag sir, and they are much experienced than me, but I think the most important thing is the keeping ourselves motivated, particularly if you are experimental biologists, and particularly if you are working in a place like India, doing good science, then you need to keep yourself motivated, highly motivated, and a hunger for the knowledge. I think these are the two important things I would say.
Yes, sir. Thank you, sir, for your wonderful advice. I will remember it.
Anybody who wants to clear anything, any questions to three of us waiting for the results, you people are most welcome to interact with three of us, right?
Okay, I had a small question to Professor Gadakar. So,
Yes.
sir, a while ago.
Please switch on your video so I can see you.
Yes, sir. So, a while ago you had mentioned that natural selection tends to maximize the fitness of one individual, but we often see that But we often see that while natural selection is maximizing the fitness of one individual, populations do evolve to become fitter. So
that's an emergent property. So what natural selection is doing is it will take the following situation. Suppose the interests of an individual are different from that of the group. Then individuals are not expected to sacrifice themselves for the group. They will try to maximize, and the net result of several individuals, each one trying to outdo the other, will be the result of the population.
Okay, so the result is just based upon the cumulative effect or emergent property.
Right. Yes. Yes.
Okay.
And
Thank you
this is
so much.
very important because a lot of people misuse the theory of natural selection, especially in the human context, to say you should all sacrifice yourself for the country or for the group or whatever. And that's not. And they say that's how natural selection works. I agree that if you want to sacrifice the country, that's fine, but don't blame it on natural selection. That's the point. Natural selection doesn't do that. You want to do it, you go ahead and do it. But many people have used this blame it on natural selection to tell people that they should sacrifice themselves. So there is this misuse of that. So we have to be very careful. Natural selection.
Okay, thank you so much, sir.
Hello,
There
sir.
is a question from Anurag. Yeah, go ahead, Anurag. Hello, Anurag, your hand is raised.
Hand is raised, but
yeah, I don't think he's unmuted.
Hanged for a bit when I started my video.
Okay.
Okay.
Oh, hi. Am I audible?
Audible, yes.
Okay, I'll just.
Yeah.
Okay, I'll
Can
turn
you
off
turn
my
off
video. I
video,
think that's
Anurag?
interfering.
No. Sometimes what happens is the connection, there will be, it could be
Okay, no
slow.
problem.
Also,
Yeah.
Okay, so my question was for Raghavendra sir. With regards to like the first question, sir, I really want to know how would you like sort of approach the experimental setup in that? first problem.
Yeah, I can now tell you what I told. My comment was when this question was revealed to me. I said this is a question that should be asked to a postdoc who has done the PhD and has some considerable experience in designing experiments. It's a very tough question. And I even told the organizers, I don't think undergrads will be able to answer this question, but I'm ready to be pleasantly surprised. So I can tell you now that I am pleasantly surprised. Most of you did a marvelous job in answering this rather difficult question. But to answer your question of how I would approach it, for example, if this was my PhD problem, then the first thing I would do is to understand what we call the natural history of the species in great detail. See, normally we understand a little bit and then we make a lot of assumptions and we start doing experiments. But the nature may be very different. And earlier, this natural history was the main thing in biology. Now we all think it's old-fashioned and we leave natural history and we do very advanced experiments. So the first thing I would do if I were doing this or if my student was going to do this, I would say spend a year, go to the field, observe lots of spiders, do small pilot experiments, study them in nature in great detail, understand the natural history of the species. Then you start designing experiments. In fact, no, even then. Then you start asking the right questions. And then when you have some reasonable questions, start designing experiments. So there are these three steps. What we often do is we don't give much importance to step 1 and 2. We start off with an experiment. That is the
Sort of like an ahistorical sort of an empiricist approach.
Natural history of, people, you may think that this is old fashioned, but it is very important. A notebook and a pencil and go to the field and make notes every day. What do you see? And then add some sophistication. Maybe you want to mark some spiders, maybe you want to move some spiders, maybe you want to manipulate something. But a lot of this trial and error, watching, observation, simple experiment, don't start with hypothesis testing. That should come much later. A lot of natural history information with simple experiments gives you a lot of insight. Then you make your hypothesis, and then you plan your experiment. Of course, you couldn't have done that for today, but in general, hypothesis, even making the hypothesis should come after considerable knowledge of the natural history of the species.
Yeah, okay. I think that answers my question. Thank you so much, sir. That was really insightful.
Any other students, if they want to interact? Gadaka sir is, Gadaka sir can answer anything on and everything. Maybe he will say not molecular biology, but he can answer. But Deepsika is experienced in microbiology and infection. I'm personally, I know genetics. I'm well trained in genetics, human genetics, Drosophila genetics. Anybody, if you people have any questions or interact with us, you can just put up your hand.
Sorry, sir. I know I just spoke, but maybe I'd think it would be
Please
really
go on,
fun
Anurag.
it would be really insightful for us if you could, you and Dipsika ma'am could maybe go through like the other questions pertaining to like your field. I'm sorry, I get if I'm making sense there, like just sort of just like do what Professor Gagatkar.
Of course, Mandala.
Yeah,
You would like to go first, Upendra?
Yeah, gut microbial thing. They are having extensive work, and I'm very happy to tell you that our own student, who did PhD from MBU, she worked with all the mothers in Singapore, pregnant mothers, and they have done extensive work. And when do the microbial infection, first colonization in the baby's gut start and everything, and they found that in the second trimester it can pass from the mother to the. It's not much. Maximum number of gut flora for the new neonatal or baby comes from the after mean, baby comes out to the vaginal things or the cesarean people lack few. Now people have know the microbiota compared to the those natural reproductions, and that paper came out just recently, 3 months back, and you people should read it, and it is open access, so they pay the money, so you people can access it freely, and it's published in Cell, right? The other thing which people have looked extensively on the gut microbiota is that autistic children and the dyslexia, how they are affected by the gut microbiota. Today we know that particularly in Australia, the whole population study was done, and people, those with the children with the autism or the dyslexia, they had Enterococcus species more. I mean, usually Enterococcus species are composed of only around 5%, but these children, they find up to 50, almost like say 55%. So there is a huge, so you can say that whether it is a cause or consequence. So people can ask that question, and people have done extensively. And today, in at least in the Australia, they are trying what you people have been talking about, the fecal transplant and the things as soon as the baby are very young. I mean, if they can cure this dyslexia and things like that. Personally, we work on most of the neurodegenerative diseases, and what we have, so far we haven't shown things, but what we have the data is that what we call as the leaky gut, right? So the gut, if they are not normal, some of the bacteria and the things, they get into the cavity, right, our body cavities, and that is the major cause for the death in the AML disease disorder, or say if you look at the Parkinsonism, Alzheimer. Today, my colleague Kiran Bhaskar, who is the neurobiologist at the New Mexico Medical College, what he has shown is that the first Alzheimer peptides are formed in the gut, and they migrate from the gut to the brain through the gut vagus nerve, right? So they are developing what they call vaccine for these gut microbial things, right? Which will make the Alzheimer plaques, right? So there are many opportunity now, and as we work, I mean, people are looking at the whole thing, right? Yeah, Deepshika, you want to add?
I will add. Thanks, Upendra. See, this is such an important topic that when you realize and understand that gut flora is present everywhere, in the sense, if you take from the insects to the mosquitoes to human to birds, so looks like that without flora there is no life, and this is now a proved factor. Now, having said that, now this is a consortium, so it is all about the diversity. If you talk about human gut, then we have roughly around 230,000 different species, and a couple of them getting added up as and when the deep sequencing is getting done, we know. Now, very interesting point is, because it is a diversity and not the number, how the diversity changes. So Pendra mentioned about the difference in the flora in the autistic individual, and it will be the same. So if you take the flora with the so-called healthy, now the definition of the healthy itself is in question. Who is an healthy individual? Healthy individual versus diabetic versus people suffering from metabolic disorders versus cancer versus any genetic disorder, you see that this entire flora has a pattern. So like monozygotic and dizygotic twin studies, they came to rescue in many of the cases when they found the first signature for the obesity. So the obese twins, monozygotic twins, dizygotic twins, a beautiful experiment where they separated the twins in different cities, found an amazing signature, and the signature came to rescue. That means the bacteroides number goes down in case of obese individual, and the firmicutes number goes up. Now, this became a pattern, and in fact, across the globe, this particular, the pattern of bacteroides being high and formicutes being low is seen globally. Same is they are trying to see any pattern in diabetes or in any of these genetic diseases. It is not only forming as a marker for the overall health quotient, but it is also something telling us much more, that we are what we eat, because the microbiota food web that the microbiota uses and then it kind of selects and enriches is everything that makes things better. So nowadays it's being used like everywhere. It's become a cliche word, like gut brain axis, gut liver axis, gut kidney axis, gut skin axis. You say any system, it is linked to the gut. Just making a point here that the flora is of utmost importance, and the food web that is harvested by what we eat, by the flora, and enriching the diversity is second important thing. So how do you maintain the diversity is a very major challenge. What is the right flora and what flora's diversity? This is, as I mentioned, it's not about number, but it is about, if you calculate the Shannon diversity index, it is about the diversity. Now the correct diversity makes the health, and this diversity changes. So it's always a question. How do I maintain my flora, and how do I know that my flora is correct? Of course, it's a very expensive experiment. Every time you can't get your flora sequenced and ask a question whether I'm undergoing a turmoil in my life because my flora is now changing, and how do I restore it? So many interesting and very beautiful questions. C-sections, that is the reason why nowadays they are not encouraging C-sections and only encouraging the normal delivery, because they figured out that in years to come with some modeling, that the prevalence and occurrence of diabetes will go much higher because of the increase in the C-sections. So C-sections, they don't get the correct flora from the mothers, that bit of that fecal matter, healthy flora, fecal matter, that when the baby passes out, they don't get it, because it's like, cut open the womb and then take the baby out. It's like a sterile, kind of a sterile baby, but now the cell paper says that you have some kind of a microbe. But still, so there are a lot of research and avenues open, but the point here is that flora is of utmost importance, be it for human, be it for mosquito. There is very interesting paper to show that if mosquito is not having that one specific flora, it is not going to transmit that certain viruses. So yeah, I think it is of utmost importance. And gut flora brings into account, and it brings in the subjects together. Now, what Professor Gadadkar talked about natural selection, and really it is so true, that how can you say self-sacrifice? It is totally not natural. It is kind of unnatural. So you cannot use the term natural selection for saying that you need to sacrifice for whatever. Similarly, the entire concept of crosstalk, the microbial ecology, as well as the mathematical modeling, all of these three subjects that you covered in this particular presentation, which was really amazing and fascinating by all 7 groups, it brings microbes, microbial ecology, microbe consortium actually brings in all these subjects together. And so many things are yet to be done. And because sometimes I feel that if we do not crosstalk, we will not be able to, in fact, tackle this situation. So yeah, very fascinating field again. And indeed, today's talk by all of you was an eye-opener.
Thanks a lot.
One, yeah.
Yes,
Can I
sir.
just
Yes, sir.
add the thing is that, I mean, the first, the person who said, I'm not trying to bring the religion here, our medical thing. The first person who said that, you should keep the gut and the liver properly, then and then you will be disease free. Do you know who said that? Anybody? You keep yourself free from the, like proper gut and liver properly, then you will not suffer from any disease, was told by Indian. And we never took it seriously. Only when the West people came and said that these microbes are doing well, that microbe is doing, then only we started now working seriously. But actually, it was Charaka who said it. I mean, 4,000 years back, he said that, you keep your gut and your liver properly, then you will not suffer from any disease. And this is for all of you, for your knowledge. The person who wrote the first paper in 2011 on the role of gut microbiota on the human physiology and the development, that paper was rejected by all the big journals, including Nature, Cell, and everything. They didn't even review it for almost like, say, 2 years. After 2 years, that paper was published in a microbial journal. The person thought that it should go in the Nature and the Cell, but none of them review it, and it was rejected from one journal to another. So keep that in mind. Journal rejection, or say, we should go back to our Indian history. We should be proud of it, and we should also work, try to establish it scientifically the way people want to do it. Yeah, we should try to do it in the best possible way with the experiment and many other hypotheses. We should try to prove it scientifically. That is what we needed today also, right? Thank you very much. And it was actually enlightenment for me to listen to all of you. I thought that you people will not be able to present it the way you all have done it. But it was just happy to just, I don't know, just listen to all of you, that we have such bright people now. I mean, right? Okay. I hope you all should not go to outside thinking that the West has got more science than the India. I mean, that is the only my concern. Okay. Right. Thank you very much.
Thank you, professors. So now we are ready with the results of the day. I would like to request Anubhav to share the screen. Yes, so I would like to announce the results today. Before announcing the results, I would like to mention that the scores were really, really close, and the judges and even us, while tabulating, like including the effects of both the session on 29th as well as today, it was very difficult to actually come to a conclusion, because the scores differed by as less as 1 point, 2 points or something of that sort. So I'd like to announce the results, and those who do not secure a place in the top 3, I'd like to say that your presentations were equally amazing. It is just that it was on some count of the other, one may have gotten a little more points than the others, but overall, all the presentations were like amazing, and we are, as those who have said the questions, we are amazed by the quality of presentations. So thank you all. Now, with this, I would like to go into the declaration of the results. We would start with the second runners-up of the day. So the second, it's a mistake on the slide. It will be second runners-up. So the second runners-up, or the third prize, is secured by Dhiren Bharadwaj, Priyanshu Jaseja, and Anshul Siwaj of Pragati Public School, New Delhi.
Congratulations.
Yeah. So congratulations, Priyanshu, Dhiren, and Anshul. Now we would like to go to the first runners-up for the event. Also, here is a mistake in the slide. It will be first runners-up. So the first runners-up for today's BioBlitz 2021 is the team from Aiser Mohali, Suhas Sehgal, Mahima Samrek, and Ananya Natarajan.
Happy congratulations, Suhas, Mahima, Ananya.
Thank you everyone.
Thank you so much. Thank you.
And now, at the end, I'd like to announce the names of the winners of BioBlitz 2021. The winners of BioBlitz 2021 is the team from Aiser Kolkata, comprising Shraman Jana, Audrija, and Tisha.
Congratulations, Shraman, Audrija, Tisha. Excellent.
Excellent.
So thank you everyone for joining us on this session. With this, I would like to request Soham to deliver the vote of thanks to the audience.
Yeah, so the debut edition of BioBlitz has finally concluded. I would like to extend my gratitude to the judges, Professor Raghavendra Gadavkar, Professor Dipshika Chakravarti, and Professor Upendra Nokthomba for sparing their invaluable time. I would also like to thank Dean UG, Professor Balaji Jagirdar, and former Dean UG, currently Dean Accounts and Finance, Professor P.S. Anil Kumar, for all their help and support. I thank Anubhav Goswami, Coordinator Chemenigma, for helping us throughout the event. I thank all our participants for actively participating in all rounds of the event. I also extend my gratitude to the participants who were a part of the pre-Pravega event and also spared their time for the final event. I thank all those who are watching the live stream of the event on YouTube. I thank the publicity team for making BioBlitz a huge success. I also thank the logistics team who ensured the smooth progression of today's event. I finally thank the BioBlitz team and the entire Pravega team for all their efforts. This would not have been possible without your invaluable support and contribution. Thank you everyone and stay safe. Jai Pravega.
Thank you, Soham. So with this, I would like to reiterate what you said. Our heartfelt gratitude to all the judges, all the participants, and everyone present here. Thanks. This was a debut event, debut edition of BioBlitz. We hope that it'll grow into an even bigger event with even better quality of questions and even better presentations in the years to come. As a coordinator, my best wishes to all the future coordinators of BioBlitz and all the future Pravega teams, and I wish them all the success in all their endeavors. With this, I, on behalf of Team BioBlitz, declare BioBlitz 2021 closed. Jai Pravega.
Jai Pravega. And on behalf of everybody, I would like to put my heartfelt thanks to the Pravega and organizing this vibrant BioBlitz program, which and technically and with these presentations, it was just an amazing experience. It was organized so well during this challenging time of pandemic through online, and I wish Pravega the very best in years to come. Thank you.
We all echo these sentiments.
Thank you, professors. So in every year when we have a real Pravega with actual judging with the judges, what we end usually is a photo session. So since that is not possible in a virtual scenario, I'd request the participants to please switch on their cameras if possible so that we can take a screenshot. So Aditya or Anubhav, can you please take a screenshot in the gallery mode? Anubhav?
So I'm the only one who is holidaying, right? Shukriyo, please.
Oh, sorry. Yeah, I myself forgot. Yeah. Thank you for reminding.
This together mode is really very nice. I could see this and click one.
So Anubhav, are you done?
Yes.
Thanks. Thanks, everyone.
Thank you.
Thank you
Bye-bye.
very much. Thank you very much. Bye.
Bye-bye.
So also logistics team, please end the live stream. Thank you, everyone. Bye-bye.
Thank you. Thanks a lot. Thank you, everyone.
Bye.